Harry, is it okay that we record your talk?
Yes, of course.
Okay. All right, so I'm going to turn off my camera.
Lawrence,
Yes.
will we have a record of the participants?
Yes.
Okay, thanks.
Good morning, everyone. Good morning in the, past the GMT line. I want to thank everyone to the GCS LAMISTAD conference. I don't know if it's our first conference, but it's the first one today. So today our first speaker is Dr. Harry Westphal, who is the director of Sirius, the Brazilian light source. So I think everyone knows Harry, or you can see his bio online, which I did not prepare myself to read this morning. But let's get right into it and let Harry take it away.
Thank you very much for the invitation and for the kind introduction, Lawrence. So can I don't know, I don't see my screen. Is it being shared?
Yes, and
yes, I see it. Yes,
I guess you are in presentation mode. Is that?
Yeah, I just want to know if you're seeing my slides or my reversal.
No, we see your slide.
I think I see your slides.
Yeah, so it's the right one. Okay. So good day for everyone, wherever you are right now in your time zone. It's a great pleasure to speak to you about our light source. So, and it's a great pleasure here to be with the colleagues that are promoting this new light source in our region that I believe is going to be a great success. And there are lessons to learn also from several light sources, and especially, I believe, from this one, which is currently the only one in Latin America. So, just before I begin, we are part of this Brazilian Center for Research in Energy and Materials. Is a large center of almost 2,000 people that hosts several laboratories besides Sirius. We have the Brazilian Biosciences National Laboratory, Brazilian Biorenewables National Laboratory, Nanotechnology National Laboratory, and a School of Sciences, an undergrad school. And we will start this next year now program for graduate school in about bioengineering. And what I want to, the points that I want to make about this talk is about societal impacts of X-ray science and the facilities that we build. And I'm going to use several examples of how we can use light sources to democratize science, basically giving access to advanced instrumentations for a group that wouldn't have this kind of access otherwise. It's a place where you can train and build capacity using interdisciplinary education through several collaborative networks and also international networks that work across borders and cultures and acts as a science as a tool for diplomacy. And through development of the facility and also development in the use of the facility, there are several spill-offs to medicine, microelectronics, advanced manufacturing, and others that I'm going to show you. And through anchoring ecosystems, linking academy, industry, and policymakers, you drive regional innovation in your region. And I'm going to show some examples of how we use this facility to drug discovery and to look at soil sciences for food safety and clean energy and environmental monitoring. And one of the major novelties now is our preparation for crisis response, basically a new facility that will allow us to look at give answers to emerging pandemics. I'm going to talk about this new lab. So my outline will be first give you Sirius at a glance, and then show a little bit of our user program and the technologies that we develop. I'm going to give a few highlights and then I'm going to show some slides about Orion, is our new project. And later I'll show a slide on some of the lessons that I think can be useful for LAMISTAD. So first, Sirius is a 3GeV machine with a natural emittance of 250 picometer radian. Is one of the fourth generation machines in the world now. I believe there are 7 in the world. It's a greenfield facility. We started construction in 2015 with a lot of the investments being done in the country itself. Now we have 27 beam lines in our portfolio, 14 of them are in operation and 13 in construction. This belongs to phase 1, phase 2, and this Orion projects that foresees 3 new beam lines. We're currently working at 200 milliamps of current, but we are ramping up next year for 350 milliamps. And our first regular user call was in November 2022. And we're offering about 5,000 hours of user beam time per year in a mature state. So this is the facility at a glance. I'm going to show a small movie about the facility, a bit of simulation. Some parts are images that we took. So basically, we start with linear accelerator of 150 MeV. And this accelerator was provided to us by Sari. And so this accelerator is inside the tunnel of Sirius and accelerates the electrons through several modules and cavities up to 150 MeV. As you can see in this simulation, we'll see the tunnel now in real image. Just 1 second. Yes, so this is a take. Of course, there's no such blue light, but this is a take inside the tunnel. And then the electrons are injected in a booster that sits into the same tunnel of the storage ring. In your right side of the screen is the booster that boosts the energy to 3 giga electron volts. So We perform this injection procedure every minute. So we work in a top-up mode with a practically transparent injection system that causes basically no distortion that can be detected in the beamlines. So every minute we ramp the current so that we keep a steady 200 milliamps. This beamlines, part of the technology development of the storage ring was to couple with the Brazilian industry of motors and generators that produced all the electromagnets in our storage ring, 6-pole, quadrupoles, and dipole, dipoles, and all the storage rings. But we rely also on these permanent magnets for high energy photons and on undulator for high brilliance. Our beamlines, we developed most of the instrumentation that we used in the beamlines ourselves. So what you see here is one part of the system, is a delta undulator that is operational in one of our beamlines for soft X-rays. And I'm going to show you one example of this beamline. So we also developed a high dynamic double crystal monochromator that you can see here from inside this simulation. It works with nanoradian stability between the two crystals. And lots of our beamlines are based on mirrors for focusing. KB mirrors, we are starting to use now lenses, but most of our beamlines work with mirrors. And this is part of the Carnaúba beamline, what you see in the simulation. Obviously, here is just a part of the simulation where you can see data being processed. So these beamlines are divided inside the storage ring. Yellow, you see, are the phase 1 beamlines. In blue are the phase 2 beamlines. In green are the Orion beamlines. The Orion and the phase 2 beamlines, they were awarded very large grants through the new program of growth acceleration of the country. And this is a budget that we are using to build these beamlines. We basically now work at 2 cycles of proposal submission per year. On average, we have a 3-to-1 oversubscription, and we are open for users worldwide. It's a double-blind distributed proposal review evaluation system that I'm going to quickly expose. And we basically have 20 to 25% of the proposals approved are coming from abroad. This is the landscape of our beamlines that we currently have. So in the columns, you see basically the energy range of the beamlines. So starting from terahertz, this is our new beamline for terahertz, tip-enhanced terahertz imaging. ARPIS beamline here, you see, this is the current stage that we are. We are exactly here. So from on the lines, you see basically the delivery year of each one of the beamline. We started with this 5 beamlines, then we had a second wave. We have now the third wave, and we are working on the 4th wave. This third wave of beamlines, the tip-enhanced terahertz, the ARPIS, the small angle scattering, and the exhaust beamlines, they were recently inaugurated by our president, Lula. He came here to the campus to inaugurate these beamlines. What you see here is a small video. He was, in fact, the first user of the small angle scattering beamline. He performed with the scientists an experiment on it was small angle scattering of nanoparticles that are used for as bacterial system. And so Lula was the first to bring the beam system into the beamline. And this is also a very important activity in this kind of light source, because you have to bring the society, you have to bring the stakeholders and the funders of the system, and to show them that this is real, this is actually what can be done, and demystify the complexities of such a system. So Lula came with lots of politicians, did the experiment. What you see here is a full experiment of small angle scattering that is being injected into the beamline. So yeah, gonna move on. We have another four waves of beamlines that we're building, and the last ones are going to be the beamlines for the Orion project that I'm going to show. So coming into democratizing science, fostering international collaboration, and training capacity building. First, this is our proposal cycles for all the beamlines. We started with the first wave of beamlines, the 6 beamlines, and then we had the other waves of beamlines. Next wave of beamlines, the current one, we are in the 7th cycle, but I couldn't put the numbers here because we're just closing numbers now. But you can see on average, we have for every 3 proposals, we're accepting 1. If we look at the international numbers, that's what we have. So every cycle there's a fluctuation, but if we look at for the last one, so there are 120 proposals accepted, 24 of which of them were coming from international scientists, and majority of them from South America and Europe, basically. We have schools for science. We provide this school. This is Ricardo Rodrigues School of Synchrotron Light. Ricardo Rodrigues was the father of synchrotron radiation in our country, the one that created the first accelerator in the entire area. And we also, every couple of years, we provide this Sync Light School. It's an international school for advanced studies in synchrotron radiation. Both of these schools are hands-on schools, so I think this is highly important for any facility that is developing a community. Now, coming to the next point, enabling technologies and driving regional innovation. So the role of technologies in the project and in the development of the country. So first, as I said, we started developing the project for this fourth generation light source in 2012. These are the first drawings when we were coming from a third generation project to a fourth generation project. And then this was designed in more detail, and this was manufactured by this company, WEG, is a motors and generators company in the country. So we partnered with them, and for them, it was a major advancement on their own capabilities. I'm going to show later some of these results. Now, also, X-ray area detectors. We started in 2015, a project for new X-ray area detectors. We entered the MEDIPIX consortium, where we learned a lot on pixel detectors, hybrid pixel detectors. Our first drawings became more mature, and finally, in 2021, we were having our first large area detectors. This is a 10 megapixel photo counting detector based on MEDIPIX. It goes inside the vacuum. This is a vacuum tunnel. And a company was created, PyTech, as a company, Brazilian company, that sells this kind of detectors now to the rest of the world. At the same time, X-ray monochromators. We learned a new technology from my partners to use systems that allowed us to go to very high stability in our monochromators, because it doesn't matter just to build a very small source of X-rays. You have to keep everything mechanically stable. So our flagship project was to build a highly stable monochromator. So we went from simulation all the way to detailed drawing, all the way to building and manufacturing. We have several of these units of high dynamic monochromator in our beam lines now operating. What I said about magnets, this is an example of WEG. So how did this spill off into other technologies for these companies? When they learned to build the magnets for Sirius, they had to improve their thermal stabilization environments and the precision die cutting of the plates that they were stamping for making the lamination of the magnets. Now, learning how to do this improved consistently the way they did motors that directly impacted in their high-performance motor manufacturing. So it's an example of how spill-offs are learning the challenges to build magnets for high-precision accelerator can improve a company manufacturing process. At the same time, the other example is this company that started doing our area detectors, PyTech. So also we had to learn how to do fine pitch ceramic design, fully automated high density wire bonding. For instance, this is a system of wire bonding that we had to develop. High density flexible PCBs. We also had to do a lot of mechanics, fine mechanics in packaging and advanced FPGA, and all of this spilled off in different projects for this company. Nowadays, they are working in prototypes for medical CT. So this is another example of how developing technologies for a synchrotron can spill off into other areas of industrial development in the country. This is a study that was done by our colleague, Professor Sérgio Salles from Unicamp, and he interviewed several of the companies that participated in the project, the Sirius project, and how they benefited. And there were several benefits that were observed by these participants. Of course, learning new technologies, acquisition of new clients, and new investments were important. But I think that the major thing was a way of thinking of improving their own technologies beyond what they were used to do for the products and to reach new markets of highly valuable products. So this is one very important aspect, and we continue development of our systems. We delivered the first one for the beamlines, but as we progress with phase 2 and the Orion project, we are developing new version of our area detectors, now based on Timepics 4. We are now developing our own ASIC for imaging, so with HIT, which is a company also belongs to the same owners of Pytech. This is a 10-micron subpixel location photo counter that we will soon have the first prototype running in the beamlines. It went to the tape out already. Now we are packaging and producing this first prototype. And also, a lot of what we learned for the mechanics of the monochromators, we applied on our experimental end system. So this is an example of sample stage that we use for a nanoprobe that allows us to reach 1 nanometer mapping stability on XY for tachography. So our aim is to reach 1 nanometer stability for tachography in this beamline. Okay, so now I'm going to show some examples of the use of science on Sirius. I'm going to basically dig into 3 examples: clean energy, agriculture, and drug discovery. So let me start with clean energy. As probably some of you know, Brazil is one of the countries that uses most of its energy from renewable resources. We have a lot of electricity coming from hydroelectrics, but also our cars run on ethanol, and we use a lot of biofuel. And the biofuel mass production, energy production in 2024, Brazil was among the leaders in the world. So, but this comes with the generation of a lot of biomass waste. So reutilizing this biomass is a key process for everyone in the world that uses this kind of energy. So the current state of art of how you break down cellulose into sugars is this so-called LPMO enzymes. These enzymes, they work into breaking sugar. This enzyme has a kind of carries a copper ion, and this copper ion is able to break holes into the structure of cellulose and to produce this disentanglement and opening up the structure of cellulose. But for this to happen, these enzymes need to be provided with electrons. They need electron donors in the right amount, and this is typically hydrogen peroxide. Now, if you give not enough, it doesn't work. If you give too much, you break the enzyme. Now, this is undergoing research in our campus in the biorenewable laboratory. And what they found out is in the waste and the soil covered with sugarcane bagasse, they found novel microbes that can produce enzymes, a novel kind of enzyme. It's called CLOC. So it's a very effective enzyme, and I'm going to show how it does. But basically, they modified genetically fungal strains to produce this enzyme, and they increased substantially the boosting of biomass saccharification to cope with cellulose recalcitration that is found in nature. So basically, they found in what is called this dark matter of enzymatic of genetic material. They found a very interesting enzyme, and now using protein crystallography, they unveiled what this enzyme does that is so special. So basically, this enzyme has 2 coppers. One copper does something similar but not equal to the LPMOs. It's a more buried oxygen copper site that allows this opening of the cellulose network. However, it also has a copper site that allows in situ oxygen peroxide generation. So it just produces hydrogen peroxide in the right amount. So it's almost like a wireless. It's carrying its own battery of electron donors to produce the reduction process. Now, this is an ongoing research in this campus. We created at CNPM a cocktail of this enzyme that is the most efficient cocktail nowadays produced, and this is becoming now an industrial project to produce in large scale. We still have a lot of research going on about how these metal enzymes, how the ions are oxidized and reduced depending on the environment. This is an example of research where we use an electrical system to probe how the electrons, as you give electrons to the system, you put current in these enzymes. It's not exactly the same enzyme, but it's an enzyme that produces a water splitting. And you can observe, using xanes, you can observe copper oxidation and copper reduction depending on the potential where the enzyme sits on. So it's kind of trying to in situ check how this ion works. Now, from structurally, beyond the molecular structure of the enzyme, you can also see how these enzymes can be packaged to produce what they have, to actually be active on, for instance, on cellulose or any other system. So when some kinds of enzymes, they produce condensates, and this can be harmful, like when they produce prions, or this can be used for improving catalysis. Now, the group dynamics of this enzyme, the collective dynamics, is extremely important for both of these systems. So using XPCS, these researchers were able to see another kind of metallic enzyme and how does, again, hydrogen peroxide makes the enzyme move from a more subdiffusive motion, arrested motion, to a more free superdiffusive motion, bursts faster than random walks. Now, this kind of collective dynamics is highly important either for investigating diseases or for using enzymes in catalytic process. So as you can see, this is a whole package of looking at different techniques on synchrotrons to understand metallic enzymes. Now, I'm moving to the second problem that I told you. This is a work that we did with this Brazilian company, agricultural company. Is the largest in the country. It's called Embrapa. And basically, they want to investigate, and they've been doing this for several years. This region of our coast is called tablelands, where you see in red. And the soil in this region has a very strange property that something like 30 to 7 centimeters underground, you have this layer of cohesive soil. So the roots cannot penetrate the soil. Basically, a root can penetrate the soil with a few megapascal, but the penetration resistance of this kind of soil is much larger than this when it's drier. So it hinders root growth. It seems to be not man-made, is of pedogenic origin, and extremely hard when dry, but when it's moist, it's friable. And it self-regenerates. So if you wet and dry cycles, you basically go from extremely hard to extremely soft. And the microscopic origin of this mechanism is still unknown. So we partnered with this company to unravel what is the mechanism behind this, because this has an enormous impact in the productivity of sugarcane in the northeast of part of the country. So if you see here the sugarcane production, the green curve, the light green curve is the average. On the southeast, we have this higher productivity, but the northeast is almost half of the productivity in the rest of the country. And this is due to because the roots cannot penetrate the soil, the sugarcane plants cannot grow much higher than a certain size, so they don't become that productive. So we started a multidisciplinary, we recruited a team to work. Instead of asking the users to submit proposals, we proposed the problem and we invited users for this challenge. Most of them are specialists in soil sciences. So then we dug samples from trenches that the Embrapa has, and we took samples from those trenches. And so what you see here is basically looking at different depths, and this is the penetration resistance that you find. So the root growth is basically penetration capability is around here, 2 megapascal. So you can see there is this layer that we call cohesive layer, where roots cannot grow. But basically, this is the same kind of soil that you find everywhere. And the question is, what is the cause? So we investigated from X-ray diffraction to see if there was a different composition on the soil. apparently the composition is pretty normal. Is basically quartz, kaolinite, and some gypsite, but in minor quantities. We looked at infrared spectroscopy with nano-infrared. These are tiny kaolinite particles. You see here the size. This is a spectroscopy line, and you can see that besides kaolinite, there's traces of calcite in these particles, which was a hint that maybe there's something connected. We don't think that necessarily this is the connection with the cohesion mechanism. But if we look at the different tomographies that we did from soil samples, you can see the porosity is highly dependent on where you are in the soil. So it seems that the pore structure in the cohesive soil is naturally different from the other parts of the soil. So we did a quantitative analysis from microtomography and on tachography, also looking at nanoporosity. And so we mapped how this porosity depended on the soil depth, and how does this connect with penetration resistance in samples measured in the lab. And there seems to be a connection with how kaolinite arranges in these different layers, especially in the cohesive layer of the soil. So we are going from all the scales down to atomic scale to the field scale, looking at multiple techniques and trying to understand the mechanism that we believe that now is going to be published. Unfortunately, I cannot show right now, but this is it's a problem now that we believe we understand the origin. Now, a second project with Embrapa is how do we solve this? We decided to separate because the second one is not only a science project, but it's also an economic, and there's a lot of impact. It's not every solution that is going to be feasible for solving this kind of problem. The third problem that I mentioned is drug discovery. So as you know, Brazil, like many other countries here in Latin America, have a huge biodiversity. So you can see the map of biodiversity, and we can find lots of different molecules that can be used as natural products that can where you can extract compounds that can be transformed in drugs. The majority of the bioactive and approved drugs that represents small molecules in the world right now. 56% of the bioactive molecules are in this sp3 space, so basically has chiral systems. And 63% of approved drugs also have this kind of chiral systems. And we have a program here in the campus that investigates molecules from this biodiversity. And basically, we start from a library, and we can do screening and confirmation using a different mass spectroscopy. And one step of this process is where they go to Sirius, where they understand novel binding sites, and they use artificial intelligence to understand the molecules that are binding in this binding site. So essentially what they do is they take tiny crystals, they take the fraction of this natural product, is I like to call it a soup of molecules, which has lots of them, and one of these molecules is the active principle. Now you soak the protein of crystals with these molecules, and some of the important molecules will bind to the site of this protein. Now, these are a few examples of molecules that are bound to the sites. Now, what they do is they do protein crystallography with these soaked crystals. Now, when you see at the electron density, you basically see blobs. How do you recognize a molecule from the blobs? Well, first, they transform these blobs of electron density in a cloud of points, and they use a deep neural network, and they take a vocabulary of generic atoms and cycles, and they look at the data set of labeled ligand images in point clouds points that there is already in the protein data bank. And basically, these 80,000 or more sites that they can label, they can then use training these networks to recognize what kind of molecule, what is the structure of the molecule that is binding to the protein. So you see, it's a different way. Nowadays, we think of artificial intelligence as a tool to recognize, to deliver the structure of a protein. Here is different. We know the structure of the protein. we want to know is the structure of the ligand. So we soak the protein with these molecules. The right one is going to, it's also a filter. The protein is also a filter for the right molecules. And then we use this blob that we take from protein crystallography. We train the neural network, and from using the artificial intelligence, the model reveals what kind of structure most likely this small molecule has. Now, we can either synthesize this in lab, or we can produce in larger quantities. And so there's a lot of work in chemistry also later, once we recognize what is the active principle of that natural product that was unveiled by by protein crystallography. So this entire program here is running as protein discovery, and it's important to notice that President Lula, when he came last time to our campus a couple of weeks ago, he inaugurated also a new building that will be responsible for taking finding these molecules closer to drugs that go to the market, so to fill in this gap with the industry. Now, last but not least, I want to show you some of the results, some of the project for Orion, which is our new laboratory. So Orion is going to be the first lab in Latin America to be able to deal with biosafety level 4 or microorganisms. Just for those who are not familiar with this language, biosafety level 4 is Ebola or Marburg or Mashupo or Sabia virus. Biosafety level 3 is SARS-CoV-2, yellow fever, West Nile, and so on. So Orion is basically this laboratory that is going to be connected to Sirius. What you see here is the building. So it's full of biosafety level 2, 3, and 4 laboratories. And all this entire region here is a region where we have the beamlines and cryo-electron microscopy. So you can bring samples from this lab and use the synchrotron technology. This is going to be the first one, the first lab in the world that is going to connect a biosafety level 4 to a synchrotron light source. Now, we are going to get from Sirius 3 beamlines. So this is boundary of the building. This is where the beamlines sit. So the first beamline is, we use the 3 beamlines to investigate 3 different scales. So the first scale is the in vivo propagation-based phase contrast tomography to look at rodents and small animals. It's going to work with a high energy range in a field of view up to 60 millimeters. So basically a field of view that you can fit small rodent and small non-human primates. To look at in vivo, so you can see in the organism how a disease evolves. Then in the smaller scale, looking at tissue by basically doing cryo X-ray tachography. So the goal of this beamline is doing virtual 3D histology. So you can look at biopsies and do virtual cuts and looking at with energy range of 5 to 20 kiloelectron volts, and a field of view of a few hundred microns to 4 millimeters in case we can use insects. In fact, insects is going to be done in this beamline. I'm going to show soon. And the resolution can run up to 50 nanometers in tachography. That's something we are projecting the beamline to do. And last, the beamline for doing in single cells. So we go from animals to tissues to single cells. This is a soft X-ray tomography beamline coupled with a cryosim, structural image microscopy, structural illumination microscopy. So basically to look at mammalian cells, bacteria, and fungi. With growing with a resolution, field of view is smaller than the other beamlines. It's going for single cells basically, and pushing resolutions down to a few tens of nanometers as well. So in more details, this is a sample preparation lab. So basically the users inside these labs, they will have the suits that are necessary for the BSL-4 environment. They can prepare samples, and in the case cryo preparation for cells and tissue. Then the users can bring these samples into the beamlines, and they couple this to the beamline system, and the samples are transferred and then measured. This is a tunnel. So all this area here is a BSL-4 area. And what we have, the beamline instrumentation is going to sit under BSL-2 environment. Now, next one is the tissue beam tomography beamline, Chimbo. So we are looking at this side of the beamline. We have a similar sample loading system. Is again a sample loading system for the beamline that allows you to bring several samples of tissue, load into vacuum. The cryo samples were prepared. And beside this, so this is an example of a brain tissue that was done with tachography in our beamline caterite. So it's this kind of image that we expect to have. And last, this previous station here is to look at insects. So what you see here is also insect tomography images that we have from the Magno beamline. So this beamline will be able to do basically insects in this end station and cells in this and tissues in this other end station. For this beamline, this detector that I showed you before, this evolution of our current area detectors is going to be used in these beamlines. Last, the small animal beamline, as I said, is going to be a beamline for propagation-based phase contrast. So it has this large tunnel where we can have the imaging detector and we use the phase contrast based on propagation. So the samples are loaded from this side. As you can see here, these are the scientists. They are loading the samples. These are normally animals. Here is the hutch where you prepare this anesthesia of the animal, and then you bring the animal into this part of the beamline. And the X-ray tomography is divided in two end stations. This one is a BSL-4 end station, and this is a BSL-3. They are separated by walls. This is the BSL-3. They have both similar preparation systems for anesthetizing and preparing the animals, but they are separated so that we can work with the BSL-3 kind of virus in vivo and BSL-4 differently. This is an example also of a kidney of a small mouse that was done on Sirius, where we can identify the structure and the counting of structures in the kidneys. So with that, I would like to conclude my talk with what I think are some very interesting lessons that we can apply to LAMISTAD as well. So first one, I think it's important that you have a user call and you open the system as soon as possible after the construction. On Sirius, we were able to open in 6 years after we start, 6 to 7 years after we start the construction. And we kept the large international quote part of the proposal cycles. This is basically, since we use a double blind system, we don't know where the proposals are coming from. But what is interesting is that this average of 20-25% of international proposals is being very interesting for the learning of our community, for establishing new collaborations. And it's not very useful for the stakeholders and the funders of a facility to understand that the facility has to be open for free for international use. But that's something that we've been always showing that this is a very interesting system to promote international collaborations. So I think this is an important lesson, keeping open for the community and start as soon as you are able to put the construction of the facility. Now, partnering with local industries to develop technology was a very important part of Sirius. At the moments where we had major difficulties, we had lots of allies in the industries that were interested in the funding in the consistent funding for Sirius coming from the government, because it was not only funding the facility, but it was funding lots of jobs that were created by these companies. So there was a whole ecosystem that was relying on the funding, not only the facility. Besides, the companies that participated on the Sirius project, they profited and they had lots of spill-offs of the technologies they learned together with us. So you can drive measurable economic returns for these companies if you use your local industry and you boost the know-how of the local industries. Now, I showed you some of the mission-oriented science, like for the soil or for biofuels or for developing drugs using our own biodiversity. And I think it's important that, of course, the facility has to be open to projects coming. On Sirius, basically, we rate the user program and we classify proposals based on their science. And the best science are the proposals that are winning typically in every call. But we also have, we understand that it's important, this is the bottom up, it's important to have the top-down approach where the facility defines with other country stakeholders some problems that have to be solved for and a strategic for regional development. And they can produce high impact results if you put together the correct group of scientists to work around this. So besides the bottom up, having top-down mission-oriented science, I think is highly important for a new facility like LAMISTAD. And something that we've done from the very early days of the other light source, the UVX, that actually brought Sirius up to life, was schools and fellowships build the user community before the facility is complete. So we had lots of training schools that were basically training the future users of the facility in the country or outside. But we needed to have a community that was specialized and could understand how to handle data, how to handle experiments, even before the facility is done. Now, phased growth is also important. You cannot build the entire facility, but you have to have plans over what you achieve in the first cycle, what you have to achieve in the next cycle. And this national funding that was dedicated for this kind of development was very important. So sustaining long-term development. Now, sustaining the maintenance of the facility is another issue, and it's I will not try to explain much how to do this because this is still a big problem in every country in the world. Inaugurating facilities is way much easier than maintaining their working. Now, I would like to thank the colleagues of LNLS that provided all the slides that I showed you, the images and the work, and that actually built this fantastic facility that we're very proud of. And the colleagues of CNPM that we work together building this campus as a vibrant science campus. And I think that the major differential that we have now is that we have across the street, we have biology labs, we have biorenewables, nanotechnology labs that can profit from Sirius, and we can profit from their know-how also in several areas that have impacted Sirius in our technologies. Finally, just to remind you, this year we have the conference, the conference XRM, X-ray microscopy conference here on Sirius in our campus. And next year we are going to have synchronous radiation instrumentation conference in Santos. We are going to have also workshops connected to Sirius, but we are going to have visits to Sirius. So we expect to have the whole vibrant community come in and join us in Brazil in the next 2 years. Thank you very much.
Thank you. Thanks for a very interesting talk. Any questions for the speaker? See, Leo, you have your hand up.
Well, Eli, thank you very much for your wonderful talk. On behalf of our project, I would like to thank you not only for the talk, but for the constant support you've given to us. I still remember that you have been the first supporter in the talk given 5 years ago in Campinas. But I would, and you have participated often in our meetings. Now I would like to pose you again a question which was posed to you by Alfonso Fuentesorias in a meeting we had 3 years ago in El Salvador. Because Alfonso asked you, do you think that a second synchrotron, there's a space for a second synchrotron in Latin America? As a matter of fact, I see that you have basically 24 proposals from abroad, part from Europe, part from Latin America, from South America. What is your perspective of a possible development of the demand which would justify a second synchrotron?
Leo, this is a very important question, but I think as I try to argue here, there are several dimensions when we are building a facility like a synchrotron light source. And of course, the scientific dimension is probably the dominant one, where you're doing a facility so that the scientists of that region can have access to this cutting edge technology. So, but it's never so clear. For instance, when Brazil started to build a synchrotron, we didn't have a large community. It was a decision on the country that we were going to build a light source and build a community. So when the first project started back in 1984, you could count in two hands the number of people that were using the synchron light source. Right? And but it was a decision to not only create a light source, but create the community. I know that there is the potential of a large community. If you look at the numbers right now, how many users we have from the Great Caribbean region into our light source, you could say, well, there's not so much, right? Even from the entire Latin America, the numbers are not so large. But that doesn't mean that people are not interested. You have to build this community. There's a lot of projects going on that would benefit from a light source, and locally having a light source makes a huge difference. Of course, you can fly over, you can fly to North America, you can fly down to Brazil, but having the light source regionally develops know-how locally also, because when you're a user of a technique, right, you use once every year or once every 2 years. Now, when you have the know-how in the country or in the region, you develop your own problems. You develop your own strategic problems. You cannot think that a strategic problem for a region is going to be accepted in any light source in the world. Take, for instance, this problem with Embrapa that we are trying to solve. We used a lot of beam time for this problem. We could never use another light source to tackle this problem, right? That would require several proposals and several synchrons. It would take way much longer to solve this problem if we were using another light source. Now, this was a problem in our country where we decided with our government, we are going to take a share of our machine to solve this problem. Now, this is something only regions that have a light source can afford to do. So even if we think about science, yes, there is a lot of potential in the community. We know that the science done in this community is potentially right for the synchrotron, and having a light source nearby is important. I think there is space for another light source, even more than one light source in our region.
Well, Ellie, of course, you know that I share what you are saying. There's no need to say, but I'm really very happy that you continue to be optimistic about the need of this infrastructure in our region. That time in Salvador, you said there's space for many more, and we are continuing to fight in this direction. Thank you very much for your support, Ellie.
Professor Capo.
All right, thanks so much. Sorry, thanks so much, Harry, for a very great presentation. And I just want to thank the organizers again for inviting me to this. Lawrence, thank you so much for including me. And I've seen a whole lot of work that is being done, from the serious side. And it's really opened up opportunities for us to be able to work together as, from various areas of the world, various continents. I'm just listening to Harry. I think there's a whole lot of collaborations that we would like to, or let me put it to say, cooperation that we would like from the African space. I'm actually speaking from South Africa, Johannesburg, South Africa, and we would like some sort of collaboration in the sense that when we have projects, sending students to Europe, to the US, it's a lot of money, that we have to spend. And sometimes having to sustain the students and, for them to learn, it's always a very big issue. But I'm just, when you were talking, I was just casting my mind back to the BRICS cooperation between Brazil, Russia, South Africa, China, and so on. That I'm just looking at, is it not possible to leverage that kind of collaborations between, say, Brazil and South Africa, and possibly the African continent as a whole, since we really do not have a light source in Africa, and try and see if we can have collaborations and, kind of interaction between us, and that we can build up collaborations and partnerships from there, which will actually open up spaces for our young students and colleagues as well, who is really interested in synchrotron science. I just want your reflection on that, and if you think that will be the way, because I didn't hear of you guys collaborating with any country from Africa, or maybe you've been doing that, but that didn't come out, from this presentation. Thank you once again for a very brilliant talk.
Thank you for your question, Professor Kapo. Yes, definitely. I think there are mechanisms on the BRICS organization to promote this kind. I understand what you're saying, that these types of collaboration, if you just sign documents is easy, but if you want to put something really to work and to bring scientists and so on, that requires funding, and this funding has to come from somewhere. But I think that BRICS has the mechanisms to do that. I know that China is active on doing this for different areas, and we actually created a virtual lab together with China recently to promote this kind of exchange. And I know that they are thinking more broadly in the BRICS system. And if you are interested, we have an area here at the CNPM campus that is responsible for this international outreach and collaboration systems. And I can direct you to them so that they have the right know-how how to reach out for the BRIC system or for any kind of funding system.
All right,
But
thank you so much.
totally
I'll look forward to that. Yeah, thanks a lot.
Any other questions for the speaker? I just, Harry, I wanted to, I have thoughts on this, on the root problem, because I spend a lot of time in my garden and in my lawn using surfactants to aerate the soil, to loosen the soil so that roots can grow deeper. So I'm going to write to you. I want to invite a full talk from that team, because I want to see what is the level of surfactants that are in the soil that you're finding, and are you having a co-crystallization problem as the soil packs?
It's not exactly that. It's much simpler actually, but yeah, we can discuss this. I mean, using surfactants, and there are several solutions that were developed along the years by Embrapa and by colleagues. But they work in a small scale. When you think about this, is an area of half of the state of São Paulo, so it's a huge area.
Gotcha.
So eventually, I mean, you can find a solution that works in the lab scale, but that doesn't necessarily work in the continental scale. So and the problem is, I'm not very optimistic on the solutions, because now we understand the mechanism is actually simple, but is hard to solve.
Gotcha. And also with Orion, I noticed you're going to have some cryo-EM machines in that space. And that's in addition to the cryo-EM machines you have in your nanotechnology area?
Yes. These are completely different new machines prepared for a BSL-4 environment. Actually, these electron microscopes, they will start operating on BSL-3 and 2. And the plan for BSL-4 and this electron microscope is more of a long term. There still needs the development. So BSL-4 will be mostly for the beamlines, but these are new cryo-EM microscopes, so that we can match, for instance, when you do cryo-EM tomography of cells, you can reach much higher resolutions than what you can do with X-rays. But sample preparation and the time it takes for doing one of those 3D images may take weeks or months. Now, in X-rays, you're aiming at statistics, so you don't want to look at one single cell and driving conclusions from there. You want to really do what we call the blobology, or looking at the blobs that define each one of the organelles and see what is significant, significative on a population of cells that has changed structurally due to the virus infection. And then once you understand that, looking at higher resolution on the electron microscope can be very, very instructive. So they kind of work complementary of each other.
Okay. Are there any questions for the speaker? Well, Harry, I'll come back to you because I did have one other question about the BSL-4 viruses. I imagine your team is looking at the Machupo virus and other sort of South American level 4 viruses.
Yeah, the team is we have a virology team that is looking at different viruses. What I know that one of their major problems right now is the Sabiá virus, which is one here in São Paulo. But yes, they acquire know-how in different areas. But the majority of the team connected to Orion is actually building the entire experimental program of this infrastructure, which is quite complicated, to be honest.
Gotcha. Okay, well, I know you got to run, so let's thank the speaker again.
Yeah.
And we'll be back in touch. I know you're very busy, but I'll try to be back in touch with you in short order.
Okay, thank you very much. I'm sorry I have to leave. There was this confusion of time zones, but I'm happy to return at any time. Thank you very much.
All right, we'll talk to you later.
Bye-bye.
Bye-bye. So I think for right now, we'll take a 30-minute break, and we'll come back to the next speaker, which will be at 11:30 Puerto Rico time. So that's 30 minutes from now.
Hello everyone. Is it on my side only, or we are yet to start? Kindly, please, if someone can help me.
They explained that we are on a break now for 30 minutes. We return in about half an hour.
Thank you, thank you, thank you. All right, thank you.
Hi, Brenda, do you want to try your slides?
Yes.
You can go ahead.
Lawrence,
Yes,
do you want me to introduce Brenda?
yes, please.
Yes. Hi, Brenda. How are you?
Hi. Good.
Good. Well, I would like then to introduce Brenda to all of us here. Brenda Lee Vargas Perez got her bachelor's degree in chemistry from the University of Puerto Rico, Mayagüez campus in 2016, in which she did research on cadmium phytoremediation under the supervision of Professor Lolita Rodriguez there. And once she got the bachelor's degree, she then moved to the Universidad Ana Geméndez Gurabo campus, in which she did a master's degree on environmental analysis, working with Professor Lisandro Kunzi, working on electrocatalytic materials for the oxygen reduction reaction. At that time, she became a sponsor by our NSF PREM, Center for Interfacial Electrochemistry of Energy Materials, the PREM program here in Puerto Rico, who is headed by the Rio Piedras campus in the University of Puerto Rico. It's a joint collaborative project with the cyclotron facility at the University of Cornell, which is CHEST, the Cornell High Energy Cyclotron Source. And Brenda's going to tell us a little bit about her introduction to chess when that happened. And then once she got her master's degree in 2021, she joined the chemistry graduate program in my campus, the Rio Piedras campus at the University of Puerto Rico. And she's currently pursuing a PhD degree in analytical chemistry there, again, working with Professor Lisandro Kunzi, now specializing in the synthesis and characterization of perovskite oxide materials, both for the oxygen evolution and the oxygen reduction reactions. And it was when she entered the PhD program in our campus that she transitioned now to this other project with CHESS, which is called the mid-scale R12, a first of its kind X-ray facility for new science on the high magnetic field, which is a project in collaboration with CHESS and the National High Magnetic Field Laboratory, MagLab, in Tallahassee, Florida, and us to construct a new high magnetic field X-ray beamline at CHESS, which is currently under construction and will contain a custom-made low-temperature superconducting magnet that's going to be generating continuous field as high as 20 Tesla. And Brenda has been involved in this project since the start. So I leave you now with Brenda to start her presentation. Brenda, take it over.
Hi, good morning, everyone. First, I would like to thank for organizing committee for the invitation for giving me the opportunity to share my experience with you. My name is Brenda Lee Vargas Perez. I am currently a PhD student in chemistry of the University of Puerto Rico, Rio Piedras campus. The presentation I'm going to share with you today is a little different from a traditional scientific start. Rather than presentation a collection of experiment results, I would like to tell you the story of how a single opportunity changed the course of my scientific career. This is the story of my journey from being in the NSF brand student to become to cyclotron user and member of the High Magnetic Field project at CHESS. Before I start with a simple clarification. CHESS is not chess. That is the part I ignore this explain opportunity. So to understand the last sentence, I first need to tell you a little about myself. Since I was sure one of my big passion was being chess. I played in tournament for many years, even organized a chess tournament myself. So for me, the word chess had always mean exactly one thing, the game. And then I started my master's degree. One day I received one email from my advisor, Dr. Lisandro Kunzi. The student line say that chess is not chess. I remember reading it, I thinking, what? Chess is chess. So I have absolutely no idea what he meant until he opened the email. Um, then short little time in our laboratory WhatsApp group, my mentor Kunzi sent another message that I still remember very clearly. Brenda, you are going to Cornell in Ferrari. That's only say that message. So I say, okay, that's okay. So at that moment, I still could not believe was happening. So, and that's was moment I really that I was about the discover to enter a new world. So I know have idea what synchrotron was. I know what chess was. I certainly never imagined that 5 years later I will become user of the facility. And today I work part to the Humanity PS project. So what is exactly chess? Before I want to introduce you for myself. So I have before I tried to share, my plan was very different from I had imagined. So first I have my bachelor degree in chemistry. Then I was science teacher. Also of my life I play share. I have 9 years until to begin my master degree. So then I know the friend project in the master program. So then I will going to my first internship in Shell in February 2019. And then I have the opportunity for the project, the Humanity Fear. After receiving that email, that's very first question I asked myself, what exactly is chess? So chess stands for the Cornell High Energy Synchrotron Source, a national research facility located at Cornell University in Ithaca, New York. It is synchrotron facility that accelerates electrons to produce a strawberry X-ray beam. We were used by research from many different disciplines to the material biological system and a wide variety of scientific phenomena. But to be completely honest, when I first arrived there, I did not fully understand the scientific background about what synchrotron. So what truly amazing with me discovered that this enormous facility was located approximately 40 feet underground and that a large portion of the synchrotron ring was actually between in one corner football field. So for someone coming from Puerto Rico, I would recently through that chess simply means the game of chess. That was something I could never have imagined. At that moment, I not have idea that this place were eventually coming to have importance in my scientific career. So until that is February 2029, I write chess for first time. I visited chess for the chess U upgrade. Was undergoing one of most significant transformation in the history the chess. This project presented a major modernization of the facility design to dramatically improve the brightness and performance X-ray beam and expanding the scientific capacity arrived to user. At that time, I was carrying out synchrotron experiment. Instead, I have the opportunity to witness firsthand the technical and engineering work behind the construction and upgrade of world-class research facility. Looking back, I realized how afortunate I was. My first experience was not only simply learning how to use a synchrotron, it's while watching is very transforming. Actually, I did full understand magnitude what I achieved that time. Those 3 months allowed to me appreciate the enormous amount of work, collaboration, and dedication required to building the scientific infrastructure that make it cutting edge research possible. So after discovery was chess, what the next step was a story very there. I was living in Puerto Rico with tropical weather and palm trees to travel to Ithaca, New York for my first internship at Cornell University. It was my first visit, yes, and honestly, everything was completely new for me, including the weather. So I have never experienced to cold temperature, snow, or anything like that before. I was entering an environment that was completely different, not only from a scientific perspective, but also from culture. I still remember arrive and see it's not everywhere. For some me coming from Puerto Rico, that's experience I will unforgettable. The only thing I need for Sentai was that I been given incredible opportunity. I wanted to learn it as much as I could. So what I did know was that the first visit were marked the beginning of a scientific journey and that GLS, our return to stress, has a synchronous user, has a graduate student working in the high magnetic field project. So my first visit, I got the opportunity to know Ken Franketti. So he's the mentor for the internship in that moment. During my first internship, I work closely connected to technical activity to work for just all great. So I have the opportunity to internship with fellow students from Puerto Rico, Hugh, Joseph, and Melissa. Now she's a good friend for me. And together we start exploring to completely new scientific working along scientific and engineering has Alan Paula. We learning how work research facility operated. Looking back, I realized that this experience was not only about science, it was also about teamwork, mentorship, and representing Puerto Rico and international user facility. During my first internship, my work was closely connected to technical activity that was part in the chef's youth upgrade. So I have the opportunity to work in different sectors, different big line, working in the monochromator, connected the part for this case. Also, as I am providing not fully understand the magnitude what happening around me. I simply knew was learning something new every single day. And looking back, I believe the experience taught me a very important lesson because every successful scientific experiment there is enormous amount of engineering, teamwork, and dedication. Also, we working in the monochromator for the something beamline half chest. So we work together with Melissa, push the pace in the monochromator, so made that the cover pace in every part in the monochromator, and Arson working in the vacuum lab. So that's very important for me because they have adopted the Brenda box. So in this box, we collocate every piece we need to check is cleaning, not have contamination. That is one responsibility I have during my internship. So looking back moment like this made feel that I was truly become part of the chess community. Then my first internship, I gone to visit a house user. In my first visit house user, we working with the camp. Also, Joseph Toto, he's in the place. So honestly, I thought my journey with chess has come to an end, but I have the opportunity to come back. During this visit, we performed an operando electrochemical X-ray absorption experiment to study energy material under real operation conditions. For this was very special moment. A few years early, I have been helping build and test part of the chess infrastructure, and now I have using the system infrastructure conductive scientific research. So there's a photography captured that moment working alongside the shop scientific staff and operating the experiment station during the data collection. That was when it truly felt I had gone from being an intern to become a synchrotron user. So Arson I had the opportunity taking training for data analysis in chess. Also visited several times for continuing operando electrochemical experiment using beamline, the peoples in here. And those experiments eventually opening the door for me to become a part of the high magnetic field project. After returning to chess several times as a synchrotron user, a new opportunity emerged that once always changed the direction of my scientific career. During my master research, I participated in the NSF Friend Program at University Ana Geméndez. Throughout that experience and collaboration established between all institutions, I had the opportunity to become involved in the new project that has beginning as the High Magnetic Fields Project. The goal of this project is developing a new beamline using high magnetic sphere along research to study material under unique conditions. For me, this represents much more than a new research project. It represents the continuation of a journey that had begun years earlier with me, very first internship. Looking back, I realized that friend did not simply introduce it to me chess. It became that ring that led me to become part of the High Magnetic Field project and eventually continue my doctoral study in the University of Puerto Rico. That moment was when I went from being chess user become part of the team helping developing new scientific capacity for this facility. And the High Magnetic Fields project aims to develop a new experimental capacity component to capacity to chess by combining synchrotron radiation with extremely high magnetic sphere using 1 magnitude 20 Tesla magnets. So this is where the sample display allowed to investigate how the structural, electronic, and magnetic property for material say under condition. For me, become part of this project means much because the simply uses synchrotron facility is the development of completely new scientific capacity to access. Merfa to the mass reward this part is raised to every started year early a simple internship opportunity. So as I continue my doctoral study, I had the opportunity to be this project. One of the things that made the experience even more special was that I was not alone. From 4 graduate students from the University of Puerto Rico have the opportunity to participate in this project. Maureen, Sebastiani, and Tanya. So, but we are participating in this project working alongside scientific, engineering, and research from chess and Cornell University. Being part of this team allowed me to see who a large scientific facility operate and who multidisciplinary collaboration make projects like high magnetic field possible. For me, this experience was much that conducted research. This was opportunity to learn, to grow professionally, and represent to Puerto Rico as part of international scientific collaboration. Also, we have the opportunity to work with Luis, Ernie, and Jacob in something tested for diffraction experiment. Also, we have the opportunity to travel to Brookhaven National Laboratory, one of the most advanced synchrotron facility in the United States. The purpose this trip was the conducting experiment on sample included in the proposal for this project. This experiment was particularly magnificent because it allowed me to see another major nationally used facility and learning how different synchrotron laboratories operate. It also reinforces the important collaboration in a scientific project. I have the opportunity not only to work the chess, but also to interaction with research and facility. Experience like this have explained both my scientific training and my purpose of the broader synchrotron community. And during this project, I have the opportunity to participate in the commissioning for tests in the new beamline. So this model is very special for me because if you imagine if when we have the beam for first time in the new beamline, also we can opportunity to work with the monochromatometer using the differential we find the refraction B in the different mirror for the monochromator at different energy. That opportunity was awesome for me. As on the commissioning time, I come to diffraction detector testing with Luis, so we can make this. This is my first. That's what my first time that made one experiment using diffraction. I never see before this experiment. So really, this is activity helped me to grow to my expecting about what is synchrotron facility. So when I look back, I realized that what started has simply interest become much more that in 2019, I said without really what a synchrotron was. I simply I have begun incredible opportunity and I wanted to learn as much as possible. Over the year, I have the privilege to return as a synchrotron user, participating in operando experiments, and then contribution to the high magnetic project beamline and working alongside amazing scientific and mentor for me. Somewhere along the way, just started beginning user reach a facility for me. It's become a place where I really have scientific, where I learning from incredible people, where I build friendship, and where I discovered the king of career I want to pursue in the future. Today, I can honestly say that she has my second home, my family, and my family, scientific family. Also, my dream is transformed to member of this just as scientific staff in the future. So also this opportunity give me the opportunity to share with my family. They stay with me during this process every time. So that's thank you for the opportunity to test the NSF project. And also I will have acknowledgement the program this possible. I am very grateful to NSF friend, CHES, the National Máquila, the University of Puerto Rico, the Universidad Ana Geméndez, my mentor, collaboration, and fellowship student had support that throughout this part. I have thank you to Dr. Lisandro Kunzi. This is my group lab now. Also, we have thank you to the Dr. Jorge Calon, Christopher Pollack, and Luis Duffer for this opportunity and for everything they can learn about when working with them. So thank you.
Very good.
Thank you.
Thank you. Thank you so much, Brenda. I think we're ready to take questions, right?
Yes.
Oh, let's see who's interesting. Anyone? Question, comment?
I have one quick question. So what is the status of the PREM grant now? Were those one of the ones that got clipped?
Brenda, you want me to answer that?
I know. We can repeat the question again because I know listen very well.
Brenda, he's asking about the PREM program, the grant, whether it's active or not. Let me just tell everybody, since I'm the PI of the project, that we got renewed for 6 more years. And so we have it until 2030. What was cut, though, was the budget. We don't have the budget for the 6 years. We were given the budget for only half of those years. So we have to stretch the budget until 2030. That's what happened, Lawrence.
Okay. We only have 958 days to go.
I know. Anybody else with a question?
Yeah, Brenda, hi, it's Ernie.
Hi, Ernie.
Wonderful presentation. Thank you so much for saying that you appreciated working here at CHES. We loved having you here at CHES. I also wanted to tell everyone who's listening that Brenda is the only other person who's trusted to operate the beamline besides the beamline scientist, Louise. And so, yeah, it'd be great to have you back. We also are waiting to get funding to operate the beamline. So there's tight funds all around. And we're still waiting for the 20 Tesla magnet to arrive, as you know. So yeah, it'd be great to have you back in Ithaca. And yeah, I guess now that you own a pair of boots, you want to come and visit us. But
so maybe Brenda can consider applying for a postdoc at Chess sometime in the future.
that'd be great.
Yeah.
Really,
The,
I have in my list.
So is the HMF actually, so it's operating, and are there results reported in the open literature already?
We're operating in a fairly constrained way, if I can say, because we don't have the 20 Tesla magnet in-house yet.
Okay.
It's these magnets are an art, I mean, you think of it as engineering and science, but it's actually an art to make it work. And so we're working closely with the vendor. I mean, we're talking with them every couple of days now. And they're struggling to deliver the magnet that meets 20 Tesla. We have confidence they will. So yeah, we're operating now doing experiments without that 20 Tesla magnet, but we can do other types of measurements.
Should we try to do a workshop on the science that you think could be revealed by photon interrogation under magnetic fields like that?
Yeah,
or do you already do that?
Yeah, I'm not the one to speak. I'm the project manager, not the scientist.
Gotcha.
So yeah, I mean, Louise and Jacob Roth have been leading the scientific effort, and I'm sure they're thinking of having workshops.
And we've had workshops in the past, and we're probably going to do more in the future. But yeah, right now we're mostly just waiting for this one piece of equipment to come in.
Is the sample environment will it accommodate samples? I guess I'm thinking of like a bird brain, or I'm looking at the sort of the problem of the bird navigation under magnetic fields.
Yeah.
So at first, the samples are going to be much smaller. There's not much room in the bore of the magnet, so a couple of millimeters, and we're going to start with powders and single crystals, and then figure out how to move to more complicated samples.
Okay. Ernie, I saw you laughed about the bird navigation problem.
Well, you know how science proposals work. You propose to do something, and then everyone thinks of many, many more things they want to do. And it's like, well, hold on, we haven't even done A. Then they want B, C, D, E, F, G, H, I. So yeah, no, but I love scientific ambition. It's great to think that we can do biology at some point. But right now, as Louise said, we just need to get one sample, millimeter sized, in the beam, and then we'll be really, really happy.
Yeah. Okay. Back to you, Jorge. I don't know if there are any other questions.
Yeah. Anybody else with any question or comment? Brenda, can I ask you something? It's about your interactions with our colleagues of the MagLab. Do you have any interactions with them, and how?
No, only see when they visit to Puerto Rico for that conference.
That we have with. Okay, good. Thank you. Well, anybody else with any question or comment? Well, can you, Brenda, just to finish up, would you just summarize very quickly the particular electrocatalysis work that you're doing now, and how you envision using the HMF to interrogate those electrocatalysts?
Okay, I work in the photosky oxide for two reaction, oxygen reduction reaction and oxygen evolution reaction. The idea is study the magnetic property this material for skyosite. Also, I using gadolinium, praseodymium, cobalt, iron. As I work other type of perovskite for oxygen that have good magnetic property using yttrium, praseodymium, lactanos. So the idea is to study the magnetic property this material. Maybe in the future we can see the correlation have the magnetic property in electrocatalysis. So that will be
Okay.
very great.
Thank you. Well, seeing no more questions from the audience, let's thank Brenda again. Work, Brenda. Lawrence, back to you.
Hi, so we're scheduled for another break for 1 hour. If you got the reminder for this conference, you know that there is a there's a webinar with the IUPAP UNESCO webinar that starts in 1 minute. It's a separate thing that's done by IUPAP and UNESCO. You can find the link on the conference website or in the email that you were sent. The speaker is Dr. Stephen Chu. I'm thinking that we're hoping that he goes for less than an hour, but for this conference, we'll reconvene in 1 hour where we have two speakers from South Africa that are both Dr. Capo's students, if I understand correctly, right?
Yes.
So, Paul, I hope you'll be back in an hour too, and can help us introduce the speakers and answer questions that they might want to throw to you instead.
Of course. Yeah, we'll do that. Thank you.
So you can look on the conference program online, and we'll reconvene in 1 hour.
Perfect. See you in an hour.
Sure.
All right. Thanks, everyone. Hi, Mary, you can try your slides now?
Okay. Is it visible?
Yes. Let me just check one thing. So we have 3 minutes. Can you indulge me? I might need another more than 3 minutes. I'll be right back.
Mary? Mary?
Okay, I'm back. Professor Capo, would you take over as chair of this session, please?
All right. Thank you so much. All right. Thank you, Lawrence. Thank you, everyone, for holding on. And thank you once again. I have the pleasure this evening of introducing the first of my PhD students, and the name is Mary George. And Mary George is a final year PhD student in my lab, molecular biophysics and structural biology lab at the University of Johannesburg, South Africa. She obtained her MSc with distinction and currently writing up her doctoral thesis, which is due for submission in October this year. She has received the prestigious ProBio Africa Research Fellowship 2 times to conduct some of her work at the ESRF in France under the supervision of Dr. Lopez. And Mary this evening will be presenting some of the work she has conducted towards a PhD using macromolecular crystallography, as well as complementing that work with biomolecular NMR spectroscopy. So Mary, the floor is yours. Take it away.
Thank you, Prof. Kapoor. Okay, so today I will be presenting on the recombinant production and biophysical characterization towards structural determination of the Aspergillus niger ring finger domain. So we all know somebody who has or had cancer, and the number of cases, new cases in 2022 stood at 20 million, and this is expected to increase by 77% by to approximately 35.5 million by the year 2050. With the increasing cancer burden, it is important, the need to find new therapeutics or more specific therapeutics is vital. Although there are various treatments available currently, such as surgery, radiation therapy, chemotherapy, hormone therapy, and targeted therapy, Zafar and colleagues in 2025 highlight the challenges that need to be overcome in order to develop effective treatment, such as targeting, combating drug-resistant cells, the side effects, and a precise targeting of cancer cells. And therefore, targeted therapy is of great interest as they look at inhibiting cancer-specific pathways or biomolecules such as proteins. This inhibition can therefore show us that proteins can be a druggable target for new anti-cancer therapeutics. And one of these proteins seen in various studies is the retinoblastoma binding protein 6, or RBPP6, which is a 250 kilodalton multi-domain splicing-associated eukaryotic protein that consists of several domains, such as the DWNN domain, the zinc knuckle domain, the P53 binding domain, the RB binding domain, and the really interesting new gene ring finger domain, which will be the focus of the study. The ring finger domain is a conserved N-terminal domain consisting of approximately 70 amino acid residues. Has conserved cysteine and histine residues, which coordinate 2 zinc ions in its core, giving the protein its stability as well as its functionality. One of the functions ascribed to the ring finger domain are that there are E3 ubiquitin protein scaffolds, E3 ubiquitin ligases that play a role as protein scaffolds in ubiquitination. Ubiquitination is a process found in cell signaling, the cell cycle, cell signaling, as well as cell death. And it does this by removing any toxic or damaged proteins. It begins with the E1, also known as ubiquitin activating enzyme, which activates ubiquitin with the help of ATP. The ubiquitin is then transferred to ubiquitin conjugating enzyme, which takes place through a thioester bond forming between E1 and ubiquitin. The ubiquitin is then transferred to ubiquitin ligase, also known as E3, through a catalytic reaction that takes place where the ubiquitin is transferred to a lysine residue found on the substrate bound to E3. This process continues until a ubiquitin chain is formed on the substrate. A signal is sent, and then the substrate along with the ubiquitin chain is sent for proteasomal degradation. A dysfunctionality in ubiquitination can cause various diseases, one of them being cancer. A paper by our group in 2023 showed us that the ring finger is involved in many cancer-related pathways such as methylation, apoptosis, and metastasis, and therefore is a attractive target for anti-cancer therapeutics. And this is also described by Copper and Colgate in 2012. However, a multiple sequence alignment study done by Copper and Colgate in 2012 showed that all the ring finger domain in all organisms are similar, except for 3. Using the human ring finger domain as search bait, they found that the Saccharomyces cerevisiae, Pichia and the Aspergillus niger had a difference in their sequence, where the Saccharomyces cerevisiae had the first and third cysteine residues missing, and the Aspergillus niger had the second coordinated cysteine residue substituted with an aspartic acid. Several ring domains several rings found in domains found in proteins have a direct implication of cancer development in human cells, and with the ring motif being conserved within species, throughout species, the mimicry between fungal proteins and human proteins is not uncommon, and therefore they can share the same immune responses and cellular processes. Therefore, the A-Niger ring can be implicated in oncogenic processes, and therefore understanding this relationship can help us gain a better understanding in its functionality. Therefore, the aim of the study was to recombinantly express and biophysically characterize the ring finger domain of the A-Niger towards structural determination using the following objectives: recombinantly expressing, purifying, and characterizing the protein, as well as setting up crystallization trials. A brief methodology that was used was transforming the protein into E. coli cells, expressing the protein in large quantities, purifying the protein using both affinity and anion exchange chromatography, then subjecting the pure protein to structural and biophysical characterization, setting up protein crystallization trials, and doing preliminary X-ray diffraction. So to begin, the PGX41 ring construct was transformed into E. coli BL21 DE3 cells, and after successful transformation, the protein was expressed in LB and induced at 0.5 millimolar IPTG, where we can see the protein, the fusion protein being expressed at approximately 37 kilodaltons. The protein was then subjected to affinity chromatography, and we can see the fusion protein being eluted successfully at 37 kilodaltons. The protein then was subjected to thrombin cleavage to separate the GST tag from the ring protein, and after successful cleavage that we can see on the gel, the protein was subjected to anion exchange chromatography to separate the tag from the protein. After successful separation, the pure protein was then run on a 10% native gel to confirm the purity of the sample. Upon collection of pure sample at good concentrations, the protein was sent for characterization, such as size exclusion chromatography, multi-angle light scattering. This was performed using a SuperDex 75 column, and the protein eluted at 13.3 moles. 99% of the mass was eluted of approximately 507.1 micrograms. This also shows us this peak, one peak showing us that the protein is a monomer. Then one component analysis was performed and found that the protein was approximately 9.9 kilodaltons, thus agreeing with Copper and Colgate in 2012, stating that the protein is 10 kilodaltons. The hydrodynamic radius of the protein was also calculated and was found to be 1.5 nanometers. And according to Verda 2017, this shows us that the protein is both globular and well-folded. Dynamic light scattering was then performed, and the protein eluted as a monomer, and it had a polydispersity index of so dynamic light scattering was performed at 4 degrees and 25 degrees. So at 4 degrees, the polydispersity index was found to be 0.345, and at 25 degrees, 0.456. This, according to Lobert 2012, a polydispersity index of 0 shows us that the protein is monodispersed, and anything up to 0.3 can be accepted, and anything above is considered polydispersed. Therefore, our sample is considered moderately polydispersed. This could be due to because of hydrophobic interactions and aggregation of the protein, and this could potentially be solved by decreasing the concentration of the protein used during sampling, as well as adding stabilizing agents. In this case, could be zinc. The characterizations following this was performed both with the apoprotein and the protein with the addition of 100 micromolar zinc chloride. So first, thermal denaturation was performed at a wavelength of 230 nanometers at a range of 20 degrees to 95 degrees. We can see a slight difference between the protein and the apoprotein and the protein containing zinc, showing a slight difference in the stability of the protein when the protein interacts with zinc. The TM of the protein was found to be 52 degrees Celsius, and of the protein with zinc at 48 degrees Celsius. This was then confirmed with a thermal shift assay, which used a Cypher orange dye where the fluorescence was measured using an absorption fluorescence of 470 nanometers and an emission fluorescence of 569 nanometers at a temperature range of 10 degrees to 100 degrees. Both samples were found to have a TM of 52 degrees. Therefore, this confirms that the protein is highly stable. Circular dichroism was then performed using a far UV range of 180 nanometers to 260 nanometers. We can see that there is a slight differences within the spectrums of the apoprotein and the protein containing zinc, and this shows us that the zinc affects the stability of the protein as well as could affect the composition of the secondary structural elements found within the protein. The Bessel server was then used to analyze the composition of the secondary structural elements found in the protein. And according to Mika 2022, the Bessel server analyzed 8 different secondary structural elements, where our protein consisted of majorly 2 secondary structural elements, where the ring protein was found to have 22.8% regular alpha helices and 30.4% parallel beta sheets, whereas the protein containing zinc had approximately 20.4% regular alpha helices and 36.5% parallel beta sheets, thus showing us that both samples had predominantly beta sheets. Thereafter, protein nuclear magnetic resonance was performed, and this is performed at 25 degrees Celsius. We can see chemical shifts downstream, 8.5 parts per million, and in the amide region, and upstream in the meta region of 1 parts per million. According to Hoffman 2005 and Pugh 2005, chemical shifts found within this area show us that the protein is well folded. However, we can see a slight change in the frequency in the amide region of the protein containing zinc, and this shows us that the protein has an interaction with zinc. We can also see the presence of alpha carbons, side chains, as well as amide protons. According to Mikhail and Krishna 2009, this shows us the presence of beta sheets within the protein. and therefore this agrees to the data we collected during circular dichromism. Thereafter, 1D hexafast was performed to determine the compactness of the protein, where both proteins had a similar compactness, showing us that the protein is both compact and well-structured. The protein was then subjected to a 1D DOSI, which measures the translation diffusion of the protein. We can see in the protein containing zinc, it has a weaker translation diffusion, therefore a higher hydrodynamic drag. Therefore, this means that the zinc interacts, or rather there's more collisions of the zinc with the protein. This, according to copper and co-workers, this therefore can implicate that the zinc is interacting with the cysteine and histidine residues found in the core of the protein. After successful characterization, the protein was then set up for various crystallization trials, and the crystals were grown using the hanging drop vapor diffusion method at 25 degrees Celsius. Two conditions obtained crystals, with one being 0.2 molar sodium fluoride, 20% PEG 3050, and 0.1 bistrous propane at pH 7.5, and the other 0.5 molar sodium fluoride and 24% PEG 3D50. Both crystals were obtained and then sent to the ID30A massive 1 ESRF beamline for testing. The crystals were found to be protein, but unfortunately did not diffract. This could be due to the protein being small and therefore unstable and flexible. Therefore, it was suggested to stabilize the protein. Therefore, we optimized the crystallization trials, and we got crystals at conditions of 0.2 molar sodium fluoride, 19-21% PEG 3050, and we added small concentrations of TCEP in order to stabilize the protein. This once again was sent to the ID30A massive 1 ESRF beamline, but unfortunately the crystals that we obtained were salt crystals and not protein crystals. Therefore, it is suggested that we continue structural determination via NMR and not X-ray crystallography. We then performed a 2D Best-Rosie NMR to see if the protein was suitable for NMR studies. And from the spectrum, we can see that the number of peaks correspond with the number of amino acid residues present in the protein, as well as that we can see the resonances in the amide region are well dispersed. This shows us that the protein is stable and well structured, and therefore can be used for NMR structural studies. Therefore, in conclusion, the protein was successfully recombinantly expressed in BL21D3 cells and purified to suitable concentrations for characterizations, where the protein was found to be a monomer at approximately a size of 10 kilodaltons and consisting of mainly parallel beta sheets. We can also see that when considering zinc and no zinc within the protein, that zinc ions are important for the protein stability, compactness, as well as folding. And therefore, this reveals to us the importance of zinc in ring finger proteins, as they are known to coordinate two zinc ions through the binding in a cross-base formation, the binding of cysteine and histidine residues. As the protein was revealed to have a substitution of a cysteine residue with an aspartic acid, these results indicate that the protein still requires zinc for its stability, and therefore can be an indicator that the protein may still serve its same function, that is ubiquitination, although further structural studies and functional assays will be needed to be performed to confirm this, and therefore lay the foundation for the design and development for new anti-cancer drugs. These are some of the references I used in my presentation. I'd just like to take this opportunity to acknowledge the University of Johannesburg my supervisors, Prof. Carpo, Dr. Masamba, and Dr. Solo Lopez, the ESRF, as well as the IBS, PSB, ISPG, PSCM for allowing me to use instrumentation and access to different characterizations for the different characterizations are performed. And finally, I'd like to thank the ProBio Africa Fellowship for funding me to go to the ESRF to perform this research, as well as the NRF for funding my doctoral studies. Thank you.
All right. Thank you so much, Mary. That was, even though you spent a bit more time than expected, I think about 1 or 2 minutes over, but yeah. So I think I would like to open this presentation up for discussion, for suggestions, questions. Yeah. So who will be the first taker? You can just unmute and possibly go out. Yes, I think that is Lawrence. Yeah. Lawrence,
I
go ahead.
wanted to ask, so I really enjoyed your talk because it did show like all the preliminary things that you have to do in the lab before you go to the synchrotron. I always say people that the going to the synchrotron is like the happy end of the beginning.
Yeah.
Or kind of an intermission in the process. But I was wondering, have your crystals suitable for cryo-EM analysis? And maybe Trevor can peep in here about what when you have a crystal that doesn't diffract in a synchrotron X-ray beamline, is a cryo-EM something to try then?
We could not use cryo-EM because I think cryo-EM takes proteins above 50 kilodaltons, and my protein is too small. It's 10 kilodaltons.
It's okay. Is Trevor on the call? Trevor, maybe Trevor would like to just say something concerning that. That'll be something his domain.
Hi there. Yes, I'm here, but I think that is a true statement. What about an NMR experiment?
Yeah, Mary. So where are you with the NMR experiment now?
So we did perform some NMR experiments, and we got some good results from it. We were able to assign the backbone and some side chains, but we still need to do more NMR experiments.
Okay. With respect to your multimers of, surely you can see them in the cryo-EM. Have you tried the elongated chains?
No, not yet. We haven't tried anything like that. I think what we've been able to do is that immediately we found out we've tried various crystals, and we have a couple of crystals which we've tried and it didn't just work out. It's either you have crystals that were beautiful but doesn't diffract, or you've got crystals that at the end of the day are just soft crystals. So we decided to go for NMR spectroscopy. Because of time constraints, we've actually done backbone assignment of the protein itself. And what we are to do is the side chain assignments. And once the side chain assignments are done, we finish doing that plus the nosy data and the backbone data. Then we take all that into Cyana and then do structural calculations. So Cyana basically automatically will generate assembles of structures, which eventually we will have to look at and then see which will now be the final model of the protein. And also, but all in all, I think what we are really interested in doing here is looking at is it really that this protein needs zinc to fold, or is zinc not needed? Then after, we can then basically try to see if we can do structure-based drug discovery using the structure of the protein. But all in all, I think that is our first question, which we want to answer by solving the structure and then looking at the 2 zinc binding sites to see if the substitution of one of the cysteine residues for aspartic acid really needs zinc to fold or not. Yeah.
Thank you.
Yeah, thanks a lot, Trevor. Thanks for that. Any other person? I'm looking at our time because of this of the other presenters. I really don't want us to. Maybe I'll take one more question from the floor again.
Quick question: if there's no others, is there a bioconjugate that you can make to give your sample more heft? So can you like conjugate it to an antibody or something like that? Well, of some known structure, for instance, that would give you more molecular weight and more. Is that an idea that was workable, Trevor, or anybody?
Yes, I think I can answer that. I think it's something that we can do. But the first thing we need to do is basically to try and look for the coordinates E1 and E2 ubiquitin enzymes. With that, we need to set up an assay. So if we can set up an ubiquitin assay from this side, and then we basically know our ring finger is an E3, we get the E2 enzyme, which has to be maybe UBCH5 or UBCH7, and then get the E1 enzyme, which is UBA2 or UBA6, I get. So if we put all three together and we're able to conduct that assay and also use gel-based systems, then we are able to tell if this protein binds, if this protein, plays a role in the ubiquitination or not. Then after the question you asked, we basically can have a chimeric protein, which basically can increase the size of this protein, possibly to maybe, make a longer construct, which fuse it with another protein. Like she mentioned that the RBP6 has some other domains as well. So we can actually fuse it possibly with the DWN domain or with the zinc finger domain, and then form a chimeric protein, which by that time we will have a bigger protein by which we can subject that to possible cryo-electron microscopy if we don't get crystals that diffract properly. So I think that is something that we can try. That's a very good advice which we can take from this presentation.
Okay, thank you.
All right. Thank you so much. Once again, Mary, thank you for your presentation. And I guess you can log off and then we'll take the next speaker. Once again, thank you for your presentation.
Thank you.
All right. Okay. So, all right, I've got the next presenter. Maybe you can go off. Let's have the next speaker. You can stop sharing.
I have stopped sharing.
Okay, now, lady, are you there?
I'm here, Prof.
Okay, can you please? Okay, perfect. Can you please just share your slides quickly? All right, okay, while that is sharing, let me quickly introduce the next speaker. The next speaker is also one of my students, a final year PhD student under my supervision. And she had an MSc as well the same year with Mary with a distinction, and currently writing up as her doctoral thesis. She received the prestigious Wood Whelan Fellowship from the IUBMB in 2024 to conduct some of her studies at Uppsala University in Sweden, and also at the Center for Medicine Discovery, University of Oxford, under the visiting professor to our department, Frank von Deft, and also spent some time at the Diamond Light Source. She, this evening, will be presenting some part of her work, which she has done, and using macromolecular crystallography toward anti-schistosoma drug discovery. So, ladies and gentlemen, join me to welcome Naledi to the platform. So, all yours, you've got 20 minutes, I guess. All right.
Okay, thank you, Prof. My name is Naledi. As Prof. Kapo has introduced me, I'm a PhD candidate at the University of Johannesburg. Just to confirm, am I audible?
Yes,
Okay,
you
thank
are.
you. So I will be presenting on some of the results on macromolecular X-ray crystallography studies of S. mansoni, CAP-DH, and the discovery and design of novel antischistosomal compounds. I have Ms. George, Dr. Grace, Dr. Kukumura, and Prof. Kapo as co-authors. In the year 2023, the Protein Data Bank reached a new milestone of having 250,000 macromolecular structures deposited. A vast majority of those structures were solved using crystallographic techniques. This makes macromolecular X-ray crystallography one of the most powerful techniques that can determine protein structures at an atomic resolution. This has now made it an essential in modern drug design, as this technique provides understanding of catalytic mechanism. It allows identification of druggable pockets and also the validation of binding modes of novel inhibitors. In today's presentation, we'll be looking into the use of macromolecular X-ray crystallography towards modern drug design and the case of schistosomiasis. Schistosomiasis is a debilitating parasitic disease, also known as bilharzia, named after Theodore Bilharz. It affects over 260 million individuals globally, with approximately 90% of those infections being accounted for in the sub-Saharan Africa, although sub-Saharan Africa only accounts for approximately 13% of the world's population, hence its association with the disease of poverty. When looking back into the continent on its own, Ghana, Nigeria, DRC, Tanzania, and Mozambique, they make the top 5 most schistosomiasis prevalent countries in the continent. There are 3 clinically common species, which include Schistosoma mansoni, Schistosoma hematobium, and Schistosoma japonicum. These parasites have a very interesting life cycle, which includes an alternate passage between the definitive human vertebrate host and the intermediate freshwater snail host. When infection in humans is inflicted when a human gets in contact with Cercaria-infested water, whereby Cercaria will penetrate the intact human skin, shed its tail and metamorphosize into schistosomelae, which will migrate to a network of blood vessels in some of the host tissues, which include the liver, whereby it matures, mates, and produces eggs, which are usually shed back into the freshwater system, where they hatch into myricidia, which will locate the intermediate snail host, therefore perpetuating the life cycle. However, it does occur that some of those eggs get entrapped inside the human host tissues, which is responsible for the resulting immunopathology. Schistosomiasis can be described as a mild infection. However, we have to take note of the fact that it does predispose its sufferers to irreversible chronic complications such as HIV or AIDS and cancer, which is very much detailed in the review highlighted above. With all of that being said, you may wonder that isn't there a drug in use? There is, praziquantal being the only sole drug that has been in consistent use for over 5 decades because it has a 60 to 90% efficiency rate. However, it is not deemed unsatisfactory due to its shortcomings, which include ineffectiveness towards juvenile schistosomes, the failure to hinder reinfection, and additionally, the exclusive consistent use has been a recipe for the emergence of drug-resistant strains that have been reported since 1999. This alone cannot overemphasize the dire need for the development of new alternative drugs. So when looking into the development of new drugs against schistosomiasis, it is very important to note that the pathology of the disease is attributable to the host immune response to continually secrete antigens into the host vasculature, meaning that the antigens that are fixated on the parasite surface have higher potential as either vaccine or drug candidates since the parasite surface is in direct contact with some of the host tissues. Studies that have investigated which antigens are fixated on the parasite surface have identified S-Mansoni GAP-DH as one of the critical antigens that actually play an important role in the evasion of the parasites into the human host, helping it to overcome biochemical and biophysical barriers, highlighting S-Mansoni GAP-DH as a molecular therapeutic target. So GAP-DH is a glycolytic enzyme which speaks to the renewed interest of metabolic enzymes as drug candidates. It catalyzes the sixth reaction of glycolysis, where there's a conversion of glyceraldehyde 3-phosphate to 1,3 biphosphoglycerides in the presence of NAD, which produces NADH that finishes electrons into the electron transport chain. These two pathways, they result in the production of ATP, which is also the reason why S-Mansoni GAP-DH is of interest, as schistosomes, like any other organisms, rely on energy generation for their survival. Therefore, this again speaks to the highlights, or rather the emphasis, of S-Mansoni GAP-DH as a molecular therapeutic target. When looking into that, we cannot never mind the fact that S-Mansoni GAP-DH is a glycolytic enzyme, meaning that it has a high conservation in nature. Nonetheless, in silico studies by the MBSB group, which I belong to, the results are not published. In addition to the structural paper published by Woreko and colleagues, it is mentioned that as much as S-Mansoni GAP-DH is highly conserved, the hydrophobic pockets along the NAD binding domain may allow differential drug design, such as the NNR region, which is highlighted in red. That then brings us to the aim of this project, which was to determine the structure of full-length S-Mansoni GAP-DH using macromolecular X-ray crystallography towards the discovery of antischistosomal compounds that can selectively bind to the NAD binding domain of S-Mansoni GAP-DH. Objectives included recombinant protein expression, purification of the protein to homogeneity, obtaining crystals with a resolution that is less than 2.5 angstrom, and solving the structure of the protein employing X-ray crystallography. Brief methodology being that we had to transform the protein into the plasmid into E. coli BL21 cells. Then selecting the positive transformants, we're gonna use them to set up protein production in TB and using AIM for autoinduction. And then after purification was conducted using a histographograph column, whereby the purified protein was then concentrated before setting crystallization trials, where we would obtain crystals which will be subjected to X-ray diffraction, which will result in the intensities of the reflection, which will allow us to get an electron density map, which allows atomic modeling and refinement to get the protein structure, or the protein model. Getting into results and discussion. In figure 7, we have results from when the conventionally synthesized gene was amplified using a Golden Gate extension. Following the protocol detailed by Michael Fairhead and colleagues, we then conducted Golden Gate cloning, and the results, or rather the positive transformants from the cloning, from subcloning and transformation into MEC1 competent cells, were screened using colony PCR, which was successful as indicated by the presence of the insect. These results were then further validated using Sanger sequencing. Thereafter, mini-prep was used to purify the plasmid, which was then used to transform E. coli BL21 cells, whereby the positive transformants we then used to set up expression in 1 liter TB MTB, that is for autoinduction. From that, the growing conditions, or rather the expression conditions, were 37 degrees for 4 hours, and overnight at 18 degrees while shaking at 200 RPM. So the following day, we would then take 1 ml to test if we do have the protein produced, with the lanes on the left, on the right, on both figure 9A and B, being proteins which are not of interest for today's presentation, meaning our interest is in the middle lanes on both figures. The lanes labeled lys as the cell lysate, sol being the soluble fraction, and LU is being the elution, amplification, and the presence of 500 millimolar imidazole. So from figure 9A, we could tell that the protein is being expressed from this lane labeled lys. However, the lane which is labeled soluble fraction indicated minimal presence of the protein in the soluble fraction. However, from the LU weight of the protein purification, we could see that the protein is actually present in substantial amounts. So this could be a result of over-dilution when the gel was being run, as protein concentration was not determined. From these results, it was indicated that the 1 liter expression does in fact produce the protein of interest. Therefore, we then proceeded to lysing the remainder of the 1 liter culture and purifying it. Hence, the presence of the band at approximately 42.2 kilodalton in figure B. Thereafter, the protein was then cleaved using TAF protease, which was successful, and then we removed the tag using reverse chromatography, hence the presence of the protein indicated by the band at approximately 36.5 kilodalton. Thereafter, it was subjected to size exclusion using the NGC, which resulted in the yield of 11 fractions indicated by the bands in figure 10B on the right. There are a presence of other bands way below 36.5, which was of interest, and this was rather validated by the presence of the shoulder and the chromatogram. Thus, moving forward, we then prioritize fraction 1 to 4 to conduct crystallization trials, which were fractionated, whereby we collected and put all the fractions together and concentrated them to 10 mg per ml before proceeding with the crystallization trials, which were then conducted using screening conditions, including the shotgun and the Morpheus screen. Within 24 hours, we were able to observe a number of crystals with different morphologies. We then fished the ones that were above 0.1 millimeter and 0.5 that were able to be carried using the loop. And then those were subjected to diffraction. And the one which is highlighted in orange, the figure in the middle, was the one which yielded a resolution of approximately 1.8 angstrom. Using this result, we then proceeded to solving the structure and subsequently optimizing the screening conditions, as our interest was into looking for inhibitors that can selectively bind to the NAD binding domain of ensmasonic gap DH. Looking into the structural dissemination and analysis, we were able to get diffraction data which shares the same space group as the one which is published by Borrego and colleagues. For interest, we were able to get a resolution which is actually higher than the one which is published. The one which is published is approximately 2.51 angstrom, and the one we were obtaining was 1.8 angstrom. In addition to that, we were able to notice that our results showed a density for the NAD binding domain, which is not observed in the published structure. In addition to that, we noticed that when we're looking into the differences between the two structures, they actually look similar, or rather they're more or less the same, other than the fact that the paper by Borrego and colleagues report on phosphorylation occurring in cysteine 153, which there's no evidence being reported on the paper. These alone would then allow a chance for a novel hollow ensmasonic gap DH structure. Moving forward into the optimization of crystallization conditions towards finding inhibitors and finding crystals which are tolerant to 0.5 0.5 to 5% DMSO. We then tried conditions of the Morpheus screen and the shotgun, where we got diffraction data of 1,9 angstrom for the crystals in figure 12A, and we got crystals of 2,24 angstrom for from crystals that we fished from figure 12B. When we tried to optimize the conditions, we then yielded crystals which are depicted in figure C. Thus, moving forward, we had to continue optimizing, mainly focusing on the precipitant concentration and the pH of the conditions which resulted in the crystals that were obtained in figure 12A. Whilst we were doing that, we also tested DMSO tolerance. This results in figure 13, being the results we were testing the tolerance of 5% DMSO. Crystals that we were able to fish from the ones in which the images are being shown in figure 13, we got results that have and diffraction data starting from 2,6 angstrom all the way to 2,77 angstrom. With those results, we then noticed that in the presence of DMSO, we're not getting a diffraction which is reproducibly below 2,5 angstrom, which is actually required if you're planning to move forward with structure-based drug design using fragment screening. We then had to go back to optimizing the crystallization conditions. With further optimizations, we were able to get better crystals in the presence of DMSO, 5% DMSO still that is. However, the results that we got from the diffraction data were from a remote session, whereby we obtained 2,11 to 2,37 angstrom resolution, which is within the range we're hoping for, meaning that we are reproducibly getting a resolution below 2,5 angstrom. However, the challenge from these results would be that this was a remote session and not an automated one, which would be the case if we were to do fragment screening. So from this result, it would be suggested that we send another batch of crystals and we do an automated collection and see if we're still getting reproducibly below 2,5 angstrom. Another challenge with the study would be the fact that phagocytic proteins, or the metabolic proteins at large, have a very high conservation in nature. And then as shown in the figure alignment, most of the differences which were being mentioned from the in silico studies and the published structure by Borrego and colleagues are mainly in the NAD binding domain, which makes it of interest instead of the catalytic domain, which is highlighted in yellow. Another suggestion would be why not just solely produce the enzymes on the NAD binding domain of GAPDH in isolation? And the answer to that would be that we have tried doing that. And from the results we obtained, it was depicted that the protein only expresses in the presence of inclusion bodies, meaning it's very insoluble, and there were little to no band in the lysate. So from this, moving forward would mean we'd have to solubilize the protein and then refold it, which is not recommended if you're planning to do fragment screening. And then another challenge would be that the NAD is bound if you're aiming for the NAD binding domain, but because this also is producing an opportunity to solve a hollow GAPDH structure, we did not have to focus on removing that. And then another thing was to try and look using a live soak test if they're actually binding pockets within the protein structure, and using that we were able to get results that actually suggest that there were 12 pockets that we can explore. And focusing also on fragment-based or rather fragment screening, another challenge would be that the targeted amino acid residues are on the surface. So with those challenges being mentioned, I would say that the concluding remarks from this study is that we can use macromolecular X-ray crystallography to get atomic level insights of biologically of biologically important targets. From this study, we were able to successfully express, purify, crystallize, and obtain high-resolution diffraction data for S-Mersonic gap DH, with the best diffracting crystals being at approximately 1,8 to 1,9 angstrom. So the aim of this study was to develop DMSO-compatible crystal systems suitable for the structure-based drug discovery, either using fragment-based screening or ritual screening. Ultimately, this work does contribute to the foundation for rational design for novel therapeutics against schistosomiasis, employing structure-based drug discovery. These are some of the references that we used to compile this study. My acknowledgements. Sorry about that. My acknowledgements with special thanks to the NRF for funding my doctoral studies and the IUBMP for funding some of my travels to Uppsala University to carry out some of the objectives. Also special thanks to CMD, which is part of the University of Oxford, whereby I did a huge chunk of the results which were presented today. Thank you.
All right. Thank you so much, Naledi. And yeah, I think taking a bit of our time, but I will still open the floor for discussion and for questions and for suggestions. So any takers? Yes, anyone on the call who would like to go? Trevor, Lawrence?
The question, I guess you don't have, did you have crystals with the drug soaked in, or in the active in the, or is the binding site of the drug itself not part of the fragment that you're trying to crystallize?
Thank you for that question. We hadn't gotten to that point yet, as the plan was to actually find the structure first and then explore either fragment screening or visual screening, as one of the challenges being that we are carrying these studies, or rather these objectives, during visits. So because of time as well, we could not really go ahead with many of the objectives. But then since the last part of the results, which I showed, we were getting closer to getting reproducibly below 2,5 angstrom. So the next objective would be to do an automated session. If we get similar results, then we would do crystallization with the drugs as well.
Okay.
All right. Okay. Any other question or comments? I will possibly take one or two more. Anyone? Because I really don't have questions for you, other than the questions discussed in our sessions during group meetings. Yeah, which I think you. Yes, Lawrence, you go ahead.
So, can you take these crystals also to cryo-EM? Are they suitable for that type of analysis?
Okay, thank you for that question as well. Because this protein of interest is a homotetramer, apologies for that, yes, it is possible to use cryo-EM for that as well. Right. So it was actually part of the discussions we were having during my visit at Uppsala University, that if time does allow, those are some of the objectives we might carry out.
I guess
can I talk a little bit? I'm sorry. I lost control of my screen for a bit and I couldn't get in.
Okay. All right. Okay, go ahead, please.
Okay. So the question I want to ask is, how different was the AlphaFold structure from the one that you determined?
Okay, thank you. The structure I kept on referring to was not an AlphaFold structure. It was actually determined using X-ray crystallography as well. So one of the two major differences that we have noticed so far is that we have a better resolution, and being also the density for the NED binding domain. So meaning that they have solved an apo structure and we would be solving a hollow structure.
But if you did you solve the structure by molecular replacement using the AlphaFold model as a starting point?
Oh no, not for the full length as Manzoni gap DH. And that was because we noticed that there's already an available structure within the PDB, which was solved using crystallographic techniques.
Okay, so you didn't, okay. And so the thing that was interesting to me then is when you take a look at the AlphaFold structure, does it significantly differ from the APO structure? And then the question then would be to make the hollow structure, whether you couldn't do that by modeling. I mean, I want you to say no, and I want you to say that there are significant differences and that AlphaFold is a mess. But I mean, we must have evidence.
I see where you are driving at, Trevor.
You see, the thing is that I mean, Africa has been taken by force along the AlphaFold track. And in fact, we're going to say that in Africa there is no more need for experimental structural determination. Have you not noticed the trend, Paul?
Yes, I get what you're saying. Yeah. I think. Sorry, I think Trevor, that's a very big discussion, which I think we can have outside this call because of our time. But I know where you are driving at. I remember the papers I sent you in the past, which talks about some of the limitations of the AlphaFold structures. If you still remember,
changed. The world has changed since then, Paul.
Yeah. Okay, we can have that chat after out of this call.
Okay, yeah, there is another thing I want to say. Is if you do all this work in Johannesburg, how much impediment is it to get the crystals to diamond for the diffraction studies? How are you managing to do that?
We basically doing the structure, everything in at Oxford, in Frank's lab over there in Oxford. And then
so you're not actually transporting crystals?
No, we're not. We do the preliminary work in Johannesburg here, knows that our protein is expressing, everything is fine, and then we send the student down to go there because we need students to learn these skills. It's a skill they basically can graduate without not having these skills. So we're not, and since we have that opportunity with Frank being a visiting professor in our department, so we then decide to use that opportunity. And it's also a learning curve for the students anyway. So, yeah.
So the future then is to actually send people to Diamond to do the entire experiment.
I guess.
What about establishing the crystallography capability and the actual career in crystallography here in South Africa?
Of course, yes, we've done that before. You know, with Priscilla, we've done that before, and that we can still build on. And we've got colleagues down the road at Wits University, the Yasin Syed group, and also we are also thinking of setting up, that facility within University of Johannesburg, and then possibly by then we can then talk about shipping samples down after we've set up the crystallizations this side. Yeah. But budget permitting, I think we did this because we've got funding for the student, and so we basically need to use that opportunity and we use the money. So yeah.
Okay.
But I quite understand what you're talking about. I think it's something which we really have to work on in establishing around here in Johannesburg. Yeah.
Okay. Thank you.
Thank you. So I guess I can't see any other person's hand up. Okay, I see something from Jojo. It's saying my video is blocked by the organizers. Is this okay? So possibly the organizers will want to
yeah,
do
it's
that.
okay.
All right.
We can we can
let
end
me
the
hand
session.
over. Yeah. Okay.
Thank
All
you.
right. Thank you so much, Naledi, and thank you for your presentation. We look forward to further engagement with you and also with every other person on this call. And Trevor, we will surely get to talk again and see what we can basically plan from here. Thank you again, Lawrence, for allowing me to share this session. And I think I have to hand over to you now.
Okay, thank you.
Yeah.
So for now, we take a 15-minute break, and then we reconvene with the final 3 talks of the day. So we'll come back in 15 minutes. And Georgia, you can just start your video, and let me also let you test your slides.
Okay, I am unable to start the video. It says that you cannot start your video because it is disabled by the host.
Okay, I'll do it right now. I'll do it. Hold on. You keep moving. Make co-host. And also to make co-host.
Okay, it's working now.
Yeah, so I normally I cut off video just to save bandwidth and to just reduce distractions from the speaker.
Okay,
So,
can I try my share?
yes, go ahead.
Okay. Okay, do you see it now?
Yes.
Fine. I will just keep it that way then until you give me the okay to start, okay? Thank you.
Right. Okay, we can get started at the last session of the day. I want to turn it over to Leo. Leo, you can introduce the speaker and moderate the session, please. Thanks.
Well, good afternoon or good evening, depending where you are. For me, it's a special pleasure to introduce Giorgio, because it is very rare that somebody can introduce a person known for 60 years. Giorgio indeed started studying in the University of Rome when I was just graduating. So I think he was a student of mine. I'm not sure, but I think he was. Then he made a very a great career, first in Italy at INFN. Then he moved to Madison, Wisconsin, and then eventually went to the École Polytechnique Fédérale of Lausanne, of which is now Professor Emeritus after having had many important positions there. And Giorgio has a tradition of working with synchrotrons, which makes him one of the people who more have contributed to the development of this instrument. And for our group, for the Greater Caribbean Initiative, it has been a great acquisition when he agreed to join our group. He's now a member of the self committee, and he has contributed a lot to the development of our project. So, Giorgio, thank you for giving this other contribution to us, and looking forward to have many more in the future. Thank you.
Okay, thank you, Leo. Can you hear me?
Yes, yes, I can.
Okay, great. Okay, so thank you, Leo, for revealing my age. But anyway, my title today.
But I also revealed mine, which is all.
Now, my title today is a bit unusual. This is not the normal topic that you talk about in a presentation for synchronous radiation, but it comes from a recent discovery of mine. And at the same time, I think it is very pertinent as far as the Caribbean initiative is concerned. So these are my coordinates. Let me go fast through them. Okay. The point is that the talk is dealing with an issue that sooner or later is going to become crucial for the Greater Caribbean synchrotron facility. And probably it is in the mind of everyone. And this issue is money. Because there are routinely, when you are proposing a big project like this, objections and questions and doubts. And so, for example, it is reasonable or not to spend a very large amount of money for one specific project, considering that the region is dramatically underfunded for research. Along the same line, I say that the questions can be dealt in a quantitative way, and I can anticipate that today I will conclude that the answer is strongly positive because of concrete facts that are not really sufficiently appreciated nor advertised until now. And the bottom line is that with synchronous radiation, we are talking about a tremendous investment, one of the most profitable investments in the world. So let's go to the discovery. First of all, I've been around this field for more than half a century, and so I could witness the progression of the field towards the present research network, which is the largest in the world. We all know the main parameters, but let me say that these figures are absolutely astonishing. We're talking about some 60 active centers, more underway. We're talking about more than 1 million Google
Giorgio,
Scholar.
are you presenting your screen? Because we don't see it, just in case.
Excuse me?
Are you presenting your slides? Because we're not seeing them.
I am able to see
I'm also
the
able to see it.
slides.
I can see the slides.
I can see the
Okay,
slides.
well, my fault. I see it now. Sorry. Go
Okay.
ahead, Giorgio.
Okay.
Sorry about
No
that.
problem, no
No
problem.
problem. Okay. So we are talking about parameters that are really gigantic. We're talking about more than 1 million Google Scholar citation. And in recent years, we have become a real factory of Nobel Prizes. Almost every year we have a few more that are coming from diverse disciplines. So all these aspects are well known, but let me go to the other aspect, that is the gigantic dimension of the financial aspect of the enterprise. So, for example, if we try to estimate the cost of the network for construction and operation, the results are a bit uncertain because the calculation methods can change. There are no general criteria, but anyway, we are talking about tens of billions of dollars. That's a big chunk of money, folks, okay? And so if you're looking at the cost of a new facility, like the Caribbean sorts, it is not going to be inexpensive. It's going to be high. And so once again, this raises question about using this money, as I already mentioned. First of all, is it wise to concentrate all this money in one single facility? Will the result be what is called a cathedral in the desert? And should the money be used instead for local research or perhaps for socially oriented projects like water or transportation or education? These are legitimate questions, but I think that should be tackled using quantitative data, okay? And we must realize that if you want to have a successful proposal for the new source, we must secure a strong support of political leaders, the research community, and the general public. And you cannot be under the illusion that you're going to get the support without good arguments. So we'll need a convincing case based on one hand on the validity from the scientific and technological point of view, but this is easy, it's well established. But also we must have a case that presents good practical results, including financial gains. And this is where the problems start, because up until now, the quantitative estimates of these gains have been extremely scarce, if not non-existent. So we are disarmed when we are trying to present the case, because everyone likes to have quantitative data and we just don't have them. Okay, so this led us to a rather timid strategy. However, good news is that this problem has just been solved, because now the financial gains from synchronous radiation can be calculated in one specific case, and we find that they are huge, beyond any imagination. And this is my main conclusion of today. So let's go. How is that the thing has been changed? Very simple, COVID. Because COVID has been such a big event that stimulated the economists to do very good, very sophisticated calculations of the cost. COVID was a tragic event, but it was also a very costly event, okay? So the economists were stimulated to calculate with great accuracy this cost. And this makes it possible then to project the results to the case of synchronous radiation. And I've been using 3 different steps in this evaluation. First of all, to identify how synchronous radiation played a role in the evolution and the consequences of the pandemics. Second, extract from the evaluation of the cost of the pandemics, which were primarily for the USA, the part that was directly mitigated by synchrotron radiation. And third, extrapolate from the results something which is valid for the entire world and for the relevant period of time. Okay, so first of all, how is that synchrotron radiation was able to contribute to the fight against COVID? We must be careful and eliminate some misconceptions. First of all, in 2020, there was the explosion of COVID, and there was a desperate need from the very beginning for drugs, but above all, of vaccines. Two levels of vaccines. One needs the atomic structure of the virus. And we all know that visible light microscopy cannot help because the wavelengths are too big with respect to the virus. So we must use X-rays, and we must use excellent sources of X-rays to shorten the analysis. And this is exactly what happened. We can see that the whole network of synchrotron X-ray sources was quickly activated, and it was incredibly fast, the process to get the first data. And this had big financial consequences that, in my opinion, largely justify the investments that have been made for more than half a century in synchrotrons. Let's go into the details. It was a very fascinating history because when COVID exploded, the first samples available were in China. And so the Shanghai synchrotron facility became almost immediately involved in the studies, specifically the two colleagues, Rao and Yang. They were able to produce the first data, but they had a big problem. The big problem is that the Shanghai facility was scheduled for a shutdown. In January 26, Rao and Yang realized that their project could not continue, and so they placed a phone call to Diamond, and Diamond was asked to take over. So they transferred the know-how, they transferred the samples and so on. And this, I believe, was an outstanding example of professional ethics and of generosity for which Rao and Yang must be praised. All right, so the victory was super fast. It took a very short time for Diamond to start producing good crystals for X-ray crystallography. And if you are making a comparison with what would have been the situation with a conventional X-ray source, you can see that the conventional source would have arrived at the same results in months, if not years. So it was very important to have synchrotron radiation involved. And of course, all the other synchrotrons were stimulated to join the fight. And so we could see here a small sample of the involved centers, but actually it was much broader than that. And it's also important to stress that no matter what was the money involved, we did stick to the common practice of making the results of synchrotron research available immediately to everyone, which allowed indeed the fast development of vaccines and drugs. Okay, I'd like to make a side note which is relevant to the case of the Caribbean source. So I've seen in the list the source in Campinas, Sirius. Sirius played a very important role. In the beginning, it just joined the efforts in order to find important structural information on the virus. But then he had a very important special result, which I mentioned here. And so he was able to make a very important contribution to this main effort. One should stress that Sirius is one of the two only synchrotron radiation facilities in the Southern Hemisphere. And it is the only one in Central and South America. And so Brazil was the only one able to contribute to this effort, which certainly was a contribution also to the good image of the country. And that's another good argument for the Caribbean source. Let me go on, okay? Because before going ahead and start making quantitative estimates, we must avoid misconceptions and try to understand what is that synchrotrons really do to fight COVID. Well, we have seen quite clearly that they were essential for the fast arrival of vaccines. We could understand that by making a comparison with the other big pandemics, the Spanish flu of 1918 and 1920. So, of course, there were no synchrotrons at that time. In fact, X-rays had started pretty recently, and the X-rays analysis with crystallography was also a novelty. And so, basically, there was no way to contribute to vaccines. The results were dramatic because the Spanish flu caused up to 100 million deaths. And even with the most pessimistic estimates for COVID, this is at least 3 times, but probably much more than were the casualties from COVID. So you can see here the concrete results from vaccines. However, we must not fall in the trap to believe that the vaccines were able to shorten the duration of the COVID pandemics, because this is not true. If you're looking at the history of the 2 pandemics, you realize that the duration was pretty much the same for both of them. So what is that the vaccines did? Well, what they did was to sharply reduce the consequences of the virus. I myself, I was ill twice because of COVID, and thanks to the fact that I had been vaccinated, I was not very ill, okay? It was a mild kind of flu. And this was the same for all the consequences, but in particular, it was very important for the premature deaths, okay? Calculations can vary, but we can guess that it was, at the very least, a reduction factor of 50%. Most likely, it was 70% or even better. For this, there is very solid experimental evidence from statistical analysis, but also from a number of targeted clinical studies, and not last, from the comparison again with the Spanish flu. So, the impact of this single consequence of the vaccines and therefore of synchro radiation was huge. Let's see, financial analysis. We started from one of the best studies on the consequences of COVID by Cutlers and Summers. Their main conclusion was that the cost for the pandemic or the pandemics for the USA and for a period of 18 months during the first phase, which lasted for 24 months, was on the order of $16 trillion. So it was a huge cost. Now, Cutlers and Summers also analyzed what were the costs that were directly related to the premature deaths, okay? And they found that there were 16 trillions. Now, if we had no synchro radiation, and therefore no vaccine, at least no fast development of vaccines, the same cost would have not stopped for the second and last phase of the pandemics. They would have continued, and the amount would have been prorated to 3.9 trillions. If you're taking the most conservative mitigation factor due to synchro radiation and vaccines, this corresponds to a saving of 1.9 trillions for the USA. How can we prorate this to the world? I propose to use the ratio of the gross national products during that period, 3.9. And so the estimated savings from synchronous radiation and vaccines was 7.4 trillions. When I got the results, I was personally shocked. 7.4 trillions is a fantastic figure, something that is beyond imagination. So we're not talking about millions, we're not talking about billions, we're talking about trillions. How is this comparing to other important figures? Well, a savings of 7.4 trillions over a period of 16 months projected over 1 year gives 5.6 trillions. And this is indeed a huge benefit. For example, it is 5% of the world gross domestic product. It is 50% of all the expenditures of the US government. And it is 15 times all the research cost within the European Union, not just the public, but also the private one. So we're talking about big money, folks. And if you are comparing this gain with the profits of the most profitable companies that are essentially Amazon, Walmart, and Apple, you find that it is almost a factor of 10 more. Now, another important parameter is what is the ratio between the profits and the investments. And once again, we had to deal with the fact that the investments are not easy to calculate because of the diversity of criteria, but basically we are talking at least 60 and maybe up to 175. So what is the conclusion? The conclusion is that we are talking about a fantastic result. Again, for comparison, this money is 22 times what the Ukrainian war is costing per year. And it is 75 times the combined yearly expenditures for armaments by Southern and Central American countries. Now, the inescapable conclusion is that we are talking about a very good investment. We must know that our evaluation of the cash benefits from synchronous radiation and vaccines are clearly an underestimate. Why so? Because we've been very, very conservative at all steps, and all the assumptions we made, the conservative assumptions and the approaches, are all working in the direction of decreasing the evaluation results. Plus, we are not talking about all the gains from synchronous radiation, but also of the small portion that is related to the studies of COVID. But there were, at the same time, many other applications of synchronous radiations, and some of them generated good benefits. I'd like to give you one example here. The Excelsius company in Switzerland has been performing for years analysis for the pharmaceutical industry. It was a very targeted study, and the result has been a decrease by several millions of the production cost and losses by the pharmaceutical companies. So we're talking about something that is very important, but I like to also to dissipate another misconception. We are not making money by selling vaccines, because if you're looking at the total volume of the sales of vaccines, it is astronomically smaller than the profits you are talking about. And so in our analysis, we could simply eliminate that from the scenes because it is totally irrelevant. Once again, we are not making money by selling drugs. Okay, what are my conclusions? First of all, we must realize that for decades, because of the lack of quantitative data on the benefits, we had to adopt a rather defensive strategy. We typically mentioned the few direct industrial uses for synchronous radiation, which are a very small part of the overall picture of the benefits. And after the discoveries that I presented today, I think that we must switch to an aggressive attitude because we are selling an extremely good product, okay? In fact, we are selling one of the best investments of all times. And so we must be proud of this result and we must advertise this result. And I also like to stress that there are a very few other examples in scientific progress of similar profits. For example, we can mention particle research such as CERN that after all produced the internet. And we could also mention the investments by Bell Laboratories in the electronic properties of semiconductors that essentially generated all the computer industry and microelectronics. So there are a very few other examples, but synchrotron radiation is a shining case. So that's my last comment, which is a famous quote by Bock and Landers. If you think that education is expensive, try ignorance. But I'd like to change this a little bit. If you think that research is expensive, try no research. Thank you very much for your attention.
Thank you. Why don't we take a few questions, Leo? Well, go ahead, Leo, since you're moderating this session.
Yes, no, thank you. Giorgio, thank you very much for your excellent talk. I remember that you anticipated something of this in the school we made in Colombia last year. Did you publish this talk? This is the first question. But the second question is that this also shows how irrelevant is what was the object of our publication in Archive about the return of investment, which is only 70% of the investment calculated over 20 years. Really, the case you published should convince any politician that this is a good investment.
It should, and the answer is not yet. I said I'm negotiating with a couple of publishing companies, and I like to make a big advertisement of this because, as I mentioned, it was a big surprise for me too. But it should become available in the next months. Okay. At the same time, I like to use all the possible opportunities to present this so people are aware of that. Okay. But once again, it is shocking. Good shock.
Leo, we see Paul Capo has his hand up.
Oh yeah, thanks, Lawrence. Thanks, Giorgio, for a very good presentation. And I really like the fact that, this speaks to science at the interface of politics and economics, if we look at it. And I think one of the things which I think we as scientists are not doing well is that we've not been able to really get these politicians and those with the keys to the vaults, for money to really support science fully like they should be. And I like your concluding statement, which speaks to that we have to be aggressive, in making sure that these guys see the need, to support this kind of investments. Yeah, and like you've mentioned, rightly mentioned in your presentation, the importance of what the synchrotron can do during the COVID pandemic. So how the synchrotron people were able to use the synchrotron, in formulating vaccine for vaccine science, and we were able all to be vaccinated. But what I want to ask you is this: what lessons do you think is there for us as scientists or researchers, or what lesson do you think is there for us, to take away from this presentation of yours so that we can, form that initiative where we speak as one voice, to these politicians, these economics, to the people that holds the key to the money. Because especially for us, from the African continent, there is no single light source within the African space. So what lessons do you think we can take from this going forward so that we will know how to speak to these guys in government? Thank you.
Okay, my personal conclusion is that we must not be shy, okay? Because, because of the weakness of our arguments until now, it was difficult to talk to politicians, okay? Because if you start saying, oh, you can use this beamline to make experiments with such and such industry, the message is lost. But if you go there and say, all right, we produce the biggest gain, one of the biggest gain in the history of the economy, okay, of all times. And we can avoid to be shy and be provocative, okay? Because the case is pretty simple. It is you just make a comparison with the Spanish flu and say you can accept 70 million people die more, which has a huge cost for humanity. It's not just a emotional cost and so on, but it is cash, okay? Because these people did not produce. These people, in some cases, were a cost to society for the rest of their lives. So this is a pretty straightforward argument. And it is based on something which is not projected, but a fact, something that happened 40 years ago, okay? Can we expect similar things in the future? You bet, okay? That is, when the next pandemic is there, we are going to have this fantastic weapon, which is our network, okay? So in a sense, it's like buying a aircraft carrier. You hope not to be forced to use it, but you better have it if it is necessary. So once again, be shy, don't be shy, and be simple. I mean, boil down the message to something extremely simple. And you can corroborate that with the other similar cases. The best one is internet coming from elementary particle research. I don't think it's sufficiently advertised that. They should.
Yeah. Thank you.
Okay. All right, thank you, Giorgio. If there's no other questions, let's move on to the next speaker. And make sure, because I got the speakers out of order the last session. Make sure I'm doing it correctly. The next speaker is Jorge Colon, who's going to talk about the Puerto Rican Light Source Initiative.
Yes.
Oh, there you are. Okay. So can you share your, yep, okay.
Okay. So, hello everybody, and good afternoon for those of you who are you in presenter mode, or are you seeing just one screen?
You can see your screen.
Just one screen. Okay, good. Okay, so I'm gonna be talking about the Puerto Rico Light Source Initiative, which is related to the Greater Caribbean Light Source Initiative, and just want to mention my co-authors, Professor Mikel Santiago from the University of Puerto Rico in Humacao, and Professor Keila Soto Hidalgo, who's from my campus in the
Leo,
Department
ho
of...
una pessima connessione, non ti sento.
How's that again?
Jorge, it is not in presentation mode.
What about now?
Now it's okay. Thank you.
Thank you. Thank you so much. Well, just to remind you that Puerto Rico is in the Caribbean, and I would encourage you to visit Puerto Rico. It's a very beautiful island. But I want to stress today that Puerto Rico used to be the site of the Arecibo Observatory, which was the largest radio telescope in the world up till a few years ago, but that unfortunately collapsed in 2020. You may have remember when that happened a few years ago. And since the collapse occurred, over the whole structure of the telescope. It was so damaged that there was no possibility of having it repaired. There were some suggestions about building a new radio telescope, but at the end, that was impossible. And what happened was that at the site, the NSF just put a proposal to build an education and outreach center, which is currently being active there. It's a collaboration between the University of Puerto Rico Rio Piedras campus, University of Maryland, and Cold Harbor Spring Laboratory, and it's nice what they're doing so far. But obviously, it's a completely different kind of endeavor. It's not doing primary science, even though it's having an important impact on the island. It's not doing top research science. So some of us have been thinking, could cyclotron source be the future big science project for Puerto Rico? Well, we've been talking about cyclotrons today in this conference. We know already about how these cyclotron facilities, by accelerating particles close to the speed of light, can generate very bright, intense beams of X-rays. The impacts, we heard a lot about them this morning. Harry from Sirius was talking about the big impacts in bioscience, medical research, environmental science, agriculture, minerals, advanced materials, engineering, forensics. So, and just now, Giorgio mentioned the Nobel Prizes that have been won by people doing research using cyclotron facilities. We know where the cyclotron sources are. Sorry. Let me take this out. Cyclotron facilities are in the US and Canada. We've been personally using the one at Cornell, the Cornell High Energy Cyclotron Sources. And as mentioned before, in this Latin American region, only the Sirius one is the one available. So how do we decide to get engaged in this facility? Well, I think it's important to remember that Puerto Rico is right there in the Caribbean. It's in what some people call the highway of hurricanes. And we got hit by a very bad one in 2017. And so dramatically reminding us of the issue of climate change and due to global warming. And we know that the effects are being felt already in Puerto Rico. There are plans to try to see how we can mitigate this, but this is affecting us already. And I've been one of the persons around the island trying to convince the government that we need to move to renewable sources of energy, engaging projects related to using the actual technologies nowadays of solar panels for Puerto Rico, but also interested in hydrogen, as we heard before about hydrogen, particularly obviously green hydrogen. So if we want to use renewable sources of energy to generate hydrogen, we need to do research because they're still expensive and they have to be improvement in, for example, electrolyzers and how to produce this green hydrogen to use them in fuel cells. And we've been involved in this type of research for some time. In my particular case, since I was a member of this NSF Center for Chemical Innovation and Solar Fuels since 2011, in which we use this type of research to also engage students in knowing more about renewable energy and the climate change issues. And then we decided to join forces with CHESS. And in 2018, thanks to the fact that Hector Abruña is a world-renowned electrochemist at Cornell University, and he's from Puerto Rico, and there were several of us who have known him for many years. His first PhD student is Dr. Carlos Cabrera. One of his postdocs was Professor Mica Santiago, and we decided to join forces in 2018, and we got this grant from NSF with the Partnership for Research and Education Materials Program and the Division of Materials Research, and we established the Center for Interfacial Electrochemistry of Energy Materials. Carlos then retired from Puerto Rico and moved to the University of Texas at El Paso, so I became the PI, and during those first 6 years, we had these research efforts in nanostructured catalyst materials for fuel cells and water splitting, also in Dyson inside solar cells, and in hybrid solid-state supercapacitors. And you can see here in the bottom center of this, of the people involved, that one of the postdocs in our initial years. Jocelyn de Pilar Albadejo became a partner in the process when she became a professor at the Maya West campus of the University of Puerto Rico. Then in 2024, we renewed the grant. We were able to get another 6-year grant. We involve other people. We have more centers in Puerto Rico involved, and we now are advancing this PREM pathway model in Puerto Rico, which I'm going to mention in a second, to look into nanomaterials for sustainable renewable energy. And so the names you see in the bottom here on the right-hand side are ones that Brenda mentioned this morning. Brenda Vargas, because Chris Pollock is one of the leaders of the People's Beamline at CHES. And Luis Dibev was a postdoc in our PREM program, and then she became a staff scientist. And the current postdoc is from Spain, Antonio Torres. And Joel Brock, who's the director of CHES, is the co-PI of this project. So the idea was, how can we help in promoting the next generation of high energy synchrotron scientists, particularly coming from Puerto Rico. So the students get recruited to the program. We work to retain them and obtain their degrees and become synchrotron scientists or scientists involved in this type of, in our case, energy-related research using synchrotron-based techniques. And you heard Brenda talk about her experience when she was involved with the upgrade of the beamlines, the storage range and the beamlines at CHES. She was able to be there for extended stay with 2 other grad students and learn the basics of how to deal with synchrotron facilities and updating the facilities. We take 4 students every year to CHES to do a summer internship program there, and they do research. For example, this student on the right-hand side was able to build a new operando electrochemical cell to do studies of this electrocatalyst there at CHES. And this was the one from last year. And each one of those get involved with summer project with scientists from CHES or Cornell. And they do a final presentation. So it's a great introduction for them to synchrotron science. So I invite you to visit our our center webpage. And some of the workshops that have been given has been put in our YouTube page, and you can see that the one on the right-hand side on fundamentals of X-ray absorption spectroscopy have been seen by more than 30,000 people already. But that relationship that we developed with CHES then evolved into a second project also with Cornell University, which was as Brenda mentioned this morning, was the high magnetic field X-ray beamline project, in this collaboration between CHES and the University of Puerto Rico Rio Piedras and the National High Magnetic Field Laboratory in Tallahassee, Florida, we got engaged in this project. And students again are involved. This is a photo on the right-hand side of one of my grad students, Tania Fernandez, involved in this project. And as Brenda mentioned, 4 students from Puerto Rico, 2 from physics and 2 from chemistry have been involved. The physics students have already graduated, and the chemistry students are still involved in the project. And we have joined commentating of their PhD projects with scientists from Cornell and Puerto Rico. And Brenda mentioned this morning about what is the high magnetic field X-ray project with installing this 20 Tesla magnet that hopefully will arrive in Cornell at a CHES sometime later this year. But we've been already, the part of the center's scientists have already been publishing the results. Experiments being conducted right there at CHES. These are some examples from the Miquel Santiago group. And in addition, Kayla Soto Hidalgo has been visiting the Canadian light source and being students there, and also have relationship with the Brookhaven National Laboratory. So we have these connections already being done. And apart from people involved with us, let me just go back for a second. Other professors more involved with nicotine receptors have been working with the argon advanced photon source to get these X-ray crystallography studies on those structures. So there's a community of users on the island who has already have relationship with synchrotron facilities. So when I learned about this initiative, about the Caribbean Light Source Project, and that was recently described in this comment in Nature 2 years ago. We got engaged, we were interested in it, and we thought, well, maybe Puerto Rico could also have an initiative of this sort. We were able to start participating in the Beyond Beams webinars that were headed by the people in Jamaica, and then met Professor Víctor Castaño and learned about the LAMISTAD initiative and the previous efforts around the Caribbean to get a cyclotron historically, and understood very well that the Greater Caribbean region is a big region, and establishing a cyclotron facility in the Greater Caribbean will also be of interest to people in the southeastern area of the US, and that all the justification why this is important is not only the one when we talk about the Greater Caribbean, it's also important for if we envision having a similar thing in Puerto Rico, as I will mention in more detail in a little bit, and how it will be something that will help develop the region in all the ways that we have been talking about today, and the financial benefits that Giorgio just mentioned is really amazing. I hope I had heard Giorgio's talk before I prepare my slides, because it gives a lot of good justification. So we know there's been several proposals throughout the years about probably establishing a new cyclotron facility in the Greater Caribbean area, Mexico, Colombia, even one in Cuba. And we've been able to gather some of those initial efforts, some strategic plans that were written and were presented before. And that's important because as we envision the possibility of engaging these type of efforts, the previous experience is very important for us. I'm sorry that all this is in Spanish. This is that case in Mexico. And important in this one, they also presented other case studies of other cyclotron facilities, including the one in Brazil, that helps to summarize what that was needed to do, and how importantly they mentioned how important was to convince the scientific community inside the island or inside the country, apart from obviously the politicians and the general public, as was mentioned before, how important it is and the time frame they took from the initial ideas to finalizing the construction projects in those facilities. It's very important, and how some efforts that have not been able to be fruitful or successful still develop this community of people interested in these ideas, how to continue working on it and create this community as the one in Mexico has been doing. So what about Puerto Rico? Well, again, as I mentioned, big science in Puerto Rico, the biggest sample of big science in Puerto Rico since the early 60s was the Arecibo Observatory, but it got destroyed in December 2020. Soon after that happened, and people heard that here in Puerto Rico, there were people interested in this Greater Caribbean initiative, and have even suggested the possibilities that, what about Puerto Rico? Could Puerto Rico be a site running parallel to the Great Caribbean project? They were interested, and they say, well, maybe we should talk about this, because it might be the next big science project for Puerto Rico. So, the authors of this talk, together with the help of Sasaki Kingwa, we come up with a white paper, a short paper which was titled "A Call for a Feasibility Study for the Construction of the Puerto Rican Light Source," and was sent to Lucy Crespos, who is the CEO of the Puerto Rico Science, Technology, and Research Trust. And we met with her a couple of times and described the possible benefits to Puerto Rico, to scientific tourism, to the development of the scientific workforce in Puerto Rico, benefits in economic benefits based on education and patents, publications, standard funding, how we increase the number of people in the workforce with careers in STEM, and that the current community, small, but the community we have in Puerto Rico have been already involved in using cyclotron facilities, will get benefit of having, obviously, a local facility. And the interdisciplinarity of the effort will be great. We have heard about that the whole day, particularly in Puerto Rico, which we have interested, obviously, in agriculture and environment, and we have had a big pharmaceutical industry on the island for many years. And the example of what happened with COVID, Giorgio explained it very well. And the impact it has in the development of new catalysts, as the ones that we are interested in renewable energy, catalysts for electrolyzers and new catalysts and cheaper catalysts for fuel cells can be developed through the use of cyclotron facilities without any doubt. But it will require a big multi-sectorial effort of all people in Puerto Rico, and how can we get people interested in the site where it can be located. We have to look not only at the location where the Arecibo Observatory was, but all around the island, and decide which site might be the best, which site will benefit the region economically in all aspects, instead of having visitors from other places that will come if this facility is ever built. The collaborative efforts, the electrical energy requirements, because as you saw after Hurricane Maria, we lost all electricity on the island. We will need to have a more robust electrical grid, and all the expansion of infrastructure that will be required with that. And we know that we, as Puerto Rico being under the jurisdiction of the United States, we will have to have participation of federal funding agencies, such as the National Science Foundation, Department of Energy, and others. We believe that we should start with a feasibility study. So we're thinking about putting a proposal together to try to get a grant to do that feasibility study. We will try to see if we can go to the federal government, or with or without the participation of the local Puerto Rican government. And it obviously will require to look into all areas of what this project will entail, and the experience that has been around the world in other facilities, and what we heard this morning from Harry about the Sirius facility will be very helpful. And now with the Giorgio recent analysis of the financial benefits of it, it will help us a lot. So we were lucky to have Galileo visit Puerto Rico. He came 2 years ago, and he gave 2 talks to students and to scientists in Puerto Rico, and he was also able to meet with Lucy Crespo, as I mentioned, the CEO of the Puerto Rico Science, Technology, and Research Trust. And during the discussion, she asked about what will be the demand for use of user facility in Puerto Rico, and what is the availability of other user beam time in other cyclotrons location. And since Puerto Rico, we've been visiting facilities in the US, we did a short investigation on that, in which we got some help from Luce DeBev from Chess, to have some statistics about the beamline facilities, talking about what is the user demand. And Joel Brock told us a little bit about how much oversubscription they have at Chess, and the challenge that we have to be able to provide the service to all those excellent proposals that get submitted to Chess. We also got contacted the Brookhaven National Lab, and also got some statistics from the beamlines to help us have a better idea of how much time is available, or how much time is not able to be given to the people asking for time. And you can see here some of the statistics. I'm going to go back for a second about the number of proposals requesting beam time and in Brookhaven, and the number of proposals that cannot be given the beam time as asked. So a lot of people cannot get beam time, and the number of users are increasing every year. So more and more users, more and more proposals, but there's not enough beam time available, which we know that there's many users in those facilities that might come from the northeast area of the United States, and they are lacking facilities in the southeast area of the United States, and they will benefit from having a cyclotron facility near that area, and it will be then an asset if there's one ever built in the southeast area of the U.S. or near it in Puerto Rico. Obviously, will also benefit the Caribbean and the Latin American region. So what we need to do? Well, we'll continue collaborating with the Greater Caribbean Light Source Initiative. We've been part of the executive committee since we started collaborating a couple of years ago, 2 or 3 years ago. In Puerto Rico, we're giving talks around the different campuses around Puerto Rico and other institutions. We've also been giving talks at the annual Senior Technical Meeting of the American Chemical Society Puerto Rico section and other scientific meetings on the area, and target to the community to get them involved in this idea, how they are interested, how they may support it, whether we collaborate in writing the proposal to do the feasibility study, how we should organize the community of users of cyclotron facilities in Puerto Rico, think about the possibility of having a symposium in Puerto Rico about cyclotron science. And then very recently, one of my undergrad students, Naomi Cruz, won an award from the American Chemical Society, because the American Chemical Society was going to send 2 students, a grad student and an undergrad student, to this workshop from the AAAS, the American Association for the Advancement of Science, to learn how to, go, about policy, how people go to Congress, to lobby Congress and people about science-related areas and how be effective. And she took that opportunity. She was selected, and she took that opportunity. And in April, she visited the office of the Resident Commissioner of Puerto Rico in the U.S. Congress. She's the only person from Puerto Rican government that participates in Congress, even though he doesn't have a vote. He does have votes within the committees. And she met with him, and I used that opportunity. I have written to him like a month or 2 before, but once she visited, and there's the photo in the middle of him with her, I contacted the office again, told them that that was my student and that we have this post-project initiative that we want them to know about, and we asked for a meeting. And when I sent them a message on Friday that we were gonna have this conference of the Greater Caribbean Light Source Initiative and that we were going to have this talk, I told them that it was happening, invited them to participate, and they called me. And the office of the resident commissioner called me, asked me for a meeting. They wanted to have it today, but it was gonna be a conflict with this talk. So hopefully, I heard that maybe it's gonna happen tomorrow. We'll see. But we want to present the idea to them, see how his office might be interested in this project and how they can help. So with that, this is very short summary of our efforts so far. We know that we are way, way behind compared to the Great Caribbean Light Source Initiative, but we're starting to get more people know about this, and we're starting to get people who had saying in government, like the Puerto Rico Science, Technology, and Research Trust. And hopefully, when we have this meeting with the Office of the Resident Commissioner, maybe we might end up with some support to do the first feasibility study, because we don't know yet what's gonna reveal and the conclusions, but hopefully it will be something that will encourage us that this might be the next big science project for Puerto Rico. So with that, thank you. I'm happy to be being able to participate in this conference, and I'm open to any questions. So thank you all.
Thank you. Leo, do you want to handle questions? Well, I'll do it. All right, thanks for your talk.
Sure.
Congratulations on all your success and your students and everything you've been doing there.
Thank you.
So Arecibo now is just what all vestiges of the telescope are just now gone.
Yeah.
Mm.
Yeah. So now they have this education center to get students involved, and they do, some research projects, at the school level, and getting teachers and students involved in scientific projects
Gotcha.
and capacity building, but it's not, high level research users facility as it was before. So we lost that, and the ideas of either repairing Arecibo or constructing a new radio telescope in Arecibo was held for a few months, but it was, not possible. NSF didn't buy the idea, and so it died.
Right, I understand. Are there any, Leo, do you have a quick question? Because we're 1 minute over.
I have a minor comment which will be discussed in more detail tomorrow. But since today there are so many African colleagues listening, participating in this activity, and there may be a volatility, and tomorrow maybe they are not there. There is one thing which perhaps Jorge, you might have added, namely the fact that Pulse, our project of the Great Caribbean, a project in Iran, a project in Uzbekistan, and the African Light Project also are trying to constitute a group which really fights for the idea of a big program of light sources for the global south. And this program has received some recognition, formal, but not very much more than that, from UNESCO. So this maybe is an important point that we should also have a kind of joint action to create conviction that it is important that this project arise everywhere with a mutual support. So, so this
Sure.
is more a message for my African colleagues because, of course, Jorge, you know perfectly well this.
Yeah, definitely. I think we all need to embrace what everybody's doing and collaborate and join forces. It's the only way around. I forgot to mention, I see Ernie Fontes right here. We have also mentioned the possibility of whether we, in the meantime, whether we got ever the synchrotron or not in Puerto Rico, that there might be the possibility of having one of the beam lines at Chess or a new one at Chess being constructed to serve this community of people in Puerto Rico and the Caribbean. So that's also things that have been talked about a little bit. Okay, thank you, everybody.
All right, thank you.
And Georgia, I need that analysis you did when you get it published. It's gonna be helpful for us. Thanks.
Okay, I was going to just make a quick comment while the next speaker gets himself ready. Are you ready, Luigi?
Hi,
Is
yes.
that the Canadian light source used to actually be at the University of Wisconsin. So with respect to a Puerto Rican light source being at Chess, it's like, history repeats itself, or at least rhymes. So that's not an unprecedented model.
Good to know. Yes.
So our next speaker is Luigi, who I'm sorry, I did not look up your bio or anything. I'm very sorry. So I'll just let you just get straight to your talk, and we'll handle it that way.
Oh, okay. Thank you. I'm going to
Okay,
share
thanks.
my screen. Okay. Okay. Yes, so thank you very much. First of all, I would like to thank the organizer of this conference for giving me this opportunity. My name is Luigi Failage from the Italian National Institute for Nuclear Physics in Frascati, specifically. And today I will talk to you about our experience with the commissioning of the STAR compact light source toward high brightness inverse Compton scattering X-ray generation. It is on behalf of the whole collaboration, both INFN and the University of Calabria, southern Italy, where STAR is installed and commissioning has started. Okay. Okay, let me get the laser. The STAR project, this is a brief sketch of the organizational schematics. It started about 10 years ago, and the project leader was Luca Serafini from INFN Milan. As you can see here, it was a big collaboration involving both the INFN Milan and the LASA laboratory in Milan and the INFN Frascati in Rome. And then into 2024, Andrea Ghigo took the national responsibility as principal investigator of the project. And then last year, I took the national responsibility up to today. It is, as I was saying, a big collaboration. I'm not gonna read all the names, but basically the big INFN groups from Milan, the LASA laboratory, and the laboratory in Frascati, basically with all the services from RF design, vacuum systems, electricity, water, magnets, etc., took this major effort in this big project. In 2021, May, the contract was signed between the University of Calabria, UNICAL, and the Italian Institute for Nuclear Physics, INFN, basically for the installation, testing, and commissioning of the upgrade of the STAR LINAC in order, which was called the STAR High Energy LINAC. So this was an upgrade of the phase 1 STAR LINAC, which started around in 2015. So the upgrade in the sense of energy-wise. So the STAR phase 1 was leading to an electron energy of about 60 mega electron volt with the STAR high energy upgrade LINAC. So we promised to achieve about 150 mega electron volt. So I will give you obviously more details about the machine later. In July 2021, we completed the detailed design report of the STAR high energy upgrade. All the installation of the machine was finished in June 2023. By December of the same year, we finished the legal commissioning and the site acceptance tests of all subsystems for the machine. And basically, we then started a 2 years maintenance and support phase, which lasted until June 2025. And at the moment, we are still in the commissioning phase in order to achieve the first X-rays in order to produce the first technical and scientific publication, and as the first stone for the X-ray machine. So this is a picture rendering of the whole hangar. and the bunker itself. So STAR is a linear accelerator, normal conducting, made out of copper. It accelerates electrons produced in a radio frequency photoinjector. The electrons are accelerated in a straight line, which is about only 10 meters long, up to an H-switching dipole magnet, which gives the opportunity to choose between two lines, what we call the low energy line in yellow and the high energy line in blue, where the electrons reach two interaction chamber points right here, where they collide with an high energy laser in order to produce Compton X-rays. And specifically in the low energy line, the X-ray energy would be up to about 70 kilo electron volt. In the high energy line, the high quality X-rays would reach 350 kV. Obviously, the energies of the output X-rays are tunable by tuning the energy easily. Tuning the energy of the electron beam. Here in the hangar, you can see all the power supply for the high current magnets, the three RF power station. We have one power station in S-band, 3 gigahertz, which feeds the RF photoinjector and the first accelerating structure. And we have two C-band RF power station at 6 gigahertz, which feed the remaining two accelerating structures in C-band. Here you can see a sketch of the laser clean room, which is hosting two lasers provided by Amplitude. One in the UV regime for the photocathodes, for the electron generation, and the IR, high power laser, for the Compton interaction point. We are also, during this commissioning phase, we are also improving some implementation, some optimization algorithm in order to make more user-friendly and easier the operation and optimization of the working points, of the many working points that we want for this machine. In particular, there is an algorithm code called Giotto, implemented by a colleague of mine, Alberto Bacci from INFM Milano, which right now it's implemented in the control system of STAR, and it basically allows us to be a control room simulator, so that by basically using the values, actual values for all the main devices like solenoids, steering magnets, the RF structures, phase shifters, and laser parameters, we can basically simulate and derive what should be the optimized trajectory of the electron beam. Here you can see, for example, the control room simulator, in particular, two virtual flags. So by exporting the measurements that we have on the YAG screens of the flags along the machines, the code can actually derive many beam parameters and tell you what you should improve in order to optimize the working point. Giotto is actually an experiment funded by the National Committee number 5 of the INFN. And so the main goal, the main reason why we implemented it in the STAR control system is to fully optimize the ICS radiation flux for all different points that one wants to have. So this is a closer look at the machine. You can see the radio frequency gun here, the high brightness electron beam source, which was provided by Radio Beam Technologies in California. One S-band 3 GHz structure, which was provided by a research instrument in Germany. Two C-band Linux 6 GHz cosine gradient RF structures, which were actually designed, tested, and commissioned by INFN itself. So it is actually a combination, so not only of private companies, industry, but also research itself. So it was a very innovative and successful contract of many stakeholders. And here you can see the two beam lines, the low energy line, phase one, the high energy line, which was part of the upgrade. And here in the straight line, when the two X-ray lines are not working, we have a beam dump in the middle. This is a sketch of the RF systems, which shows how the RF power is distributed from the three klystrons, the S-band 1 and the two C-bands, which are located in the hangar, and all the way to the bunker area. And basically, what we have here are a distributed number of vacuum pumps. There is a circulator isolator, which is typical for a standing wave structure, while the other three sections are traveling wave structures, so they don't require such devices. You will see some standard devices like RF windows and directional couplers, which we use, and I will show how we can see the RF signals along the whole machine. This is a picture of the radio frequency gun, which, as I said, was designed, developed, and installed by Radio Beam Technologies. It is now operational. This is a picture of the gun right after brazing at SLAC laboratory. This is the first picture of the installation inside the STAR bunker. It was September 2016. This is a picture from another angle, also with the solenoid. And below you can see a beat pool measurement of the electric field on axis. So it's a low RF power measurement, which shows that actually the electric field was optimized and tuned at the right frequency. Here you have a picture of from the bottom, so starting from the gun all the way up towards the interaction points. The UV laser typically uses a Gaussian pulse with a sigma of about 1 picosecond, but it is variable up to about 5 picosecond full width of max. And we can actually extract a beam charge of a few hundred picocoulomb up to 500 picocoulomb. And the repetition rate, the radio frequency repetition rate of the machine can be up to 100 Hertz. So this is a picture inside the bunker. You can see the gun on the right, followed by the solenoid. You can actually see here also the cooling, the chiller devices, which we use to temperature stabilize all the accelerators. So we don't use the main plant. Basically, we use the main plant for magnets and other less sensitive stuff. But for the accelerator structures, we use very precise temperature stabilized chillers. You can see the S-band structure right here. and the 2 Siemens Linux following. And this is a picture from the other side. So basically you have the linear accelerating section coming from here, which is produces an electron beam. So as I said, from with tunable energy from 60 MeV up to 150 MeV. You can choose to direct the beam either to the right, to the low energy line, or to the left, to the high energy line in the Compton indirection point chambers. Briefly on the magnetic elements, we have many of them from trajectory correctors, steering magnets. We have the H switching magnets, which is for us one of the most important because it allows us to choose the beamline for the electron beam. And then we have the 2 dog legs or transfer lines with their own quadrupole magnets in order to adjust for the dispersion due to the curvature of the beamline going from the straight line to the line of the interaction chambers. And eventually, after the interaction, the electron beam, after generating the X-rays, it is dumped downward to the floor by using big magnets, like especially for the high energy line, we use a 90 degrees magnet, which is pictured right here, which we call the dipole beam. And so basically you can see the picture of all magnets which are actually installed right now along the machine. Beam diagnostics, we have beam position monitors, beam current monitors, YAG screens, which are the YAG screens are basically also we call them flags. They are scintillators, so they allows us to see basically the electron beam in order to measure the spot size, and we also use the magnet itself, the H magnet, in order to measure the energy of the beam. So we also use it as a beam spectrometer, which is very efficient and, for my experience, the most reliable way, once we are able to accelerate, is the most reliable way to know exactly the energy of the electron beam at the end of the straight line. We use 2 Libera for the diagnostics. We use some Libera beam position monitor processing modules, which were provided by instrumentation technology. So the control room, basically we started the phase 2 upgrade, as I said before, in 2021. This is a picture of the very initial control room, which we were using at that time. And after, basically this is from last year, a picture from last year. So the group grew, and we were able to move away from the hangar area, so everything is remotely controlled, and timing and the control system basically was optimized, and we do everything remotely today. Talking about the control system, I will just, it's very complicated system for us. It was the first time to use the EPICS architecture, the experimental physics and industrial control system framework, which is a software infrastructure for building distributed control systems. And this is a very robust and reliable framework which is used today in major accelerator system facilities. Thanks to EPICS, we are able to handle all the subsystems of the machine from the low-level radio frequency, vacuum, magnets, beam position monitors, camera, etc. As operator interface, we use the program Phoebus, which is really user-friendly. In this picture, I only took one example of the Phoebus display for the low-level radio frequency subsystem of the three klystron, K1, K2, and K3. And here is a picture of the main radio frequency signal from the gun, from the klystron forward you see here, from the gun, the beam source itself. And this is the first accelerating structure. So we're able to control the radio frequency signal in each point of the machine thanks to this software. Here, it was the first day that we actually saw the first electron beam. It was July 2025. And here you can see on the oscilloscope how we synchronized the UV laser on the photocathodes. And here we were measuring the first electron beam by looking at a Faraday cup. This was the first Faraday cup downstream of the electron gun. So the first measurement was actually we were using a laser of about 10 picoseconds, and we measured a charge of about 100 picocoulomb. So this was the very first measurement. Then we started to optimize also by the help of the Giotto software, which I talked before about. This is a picture of the electron beam. This is already more optimized and uniform. It's soon after the solenoid. This picture is from the camera of one of the YAG screen, the first flag after the solenoid of the gun. We then transported after the first S-band LINAC. This is another picture from the flag after the LINAC. And eventually we transported all the way to the interaction chamber, where we can actually use the quadrupole magnets in the transfer line in order to obtain the sigma, the spot size of the beam that we like. In this picture, for example, it was about 0.3 millimeter. We can change this size. It's very simple. So this is another flexibility of working with this linear accelerator by playing with the power, the current, so the strength of the quadrupole magnets along the transfer line, but also after the transfer line. So it is very flexible. How do we do timing and synchronization? So as I said, we have two lasers. Basically, for the UV laser and the accelerating RF structure, we use an oscilloscope. For the synchronization between the UV laser and the RF gun, we use the low-level radio frequency at the picosecond level, and we do this also by using the signals on the oscilloscope. And for the gun, we use the radio frequency signal from one of the directional couplers, both for the radio frequency gun, but also for the C-band LINACs. And we use a photodiode for the UV laser. And in this picture, you also see in red is a plot of one of the beam position monitors. And here I try to summarize the main signals that we use, for example, for a temporal synchronization at the interaction point, which I will show then how we do it physically. We use an ultra-fast gigahertz level for the IR laser, which can reach 500 millijoule and 5 picosecond full width max. This is the UV laser, so basically it tells you what is the timing of the electron generated at the source. And here is the beam position monitor for the electron beam soon after the interaction point. So what we do is to bring the signal very close at the nanosecond level on the oscilloscope, like in this case, and then we start scanning the IR laser, the high power laser, through the beam with very small steps of below picosecond level steps. So it takes time, but it is very simple with this flexible IR laser to scan and sweep along the electron beam to get the synchronization and X-ray generation. And this is a picture of what I showed before. This is how we do it in practice. We monitor the electron beam in the BPM soon after the interaction point, which is right here. And we use a fast IR photodiode for the high energy laser, which comes from here and goes to the interaction point. And we monitor the signals on the oscilloscope that I showed before. So I want to show you, let's see if it works. This is a movie, one of the first one, where you can see the laser spot on the YAG screen and this fading one, the less intense, and the intense beam is the electron beam up to about 100 MeV, and they were being adjusted related to each other. So we were actually, by keeping the IR laser fixed, we were moving the electron beam in order to get a perfect a spatial match, so spatial superposition on the YAG screen. Obviously, in this measurement, the power of the laser was much lower. So it was in the range of a few hundred. If it was in the microjoule range, otherwise the YAG screen would break. So the idea is to align them in space with the YAG screen at low power for the IR laser, and then get rid of the YAG screen and increase the IR laser energy full to maximum, 500 millijoule, and to the right spot down to about 50 micron, which is the nominal operation point, and then start the synchronization scanning steps. How? Okay, so how do we know if we get X-rays then? So we set up a small detector station right after the interaction chamber. I'll show you here. Blue is the color of the dumping magnet, so it's where the electron beam is dumped downward to the floor. So the idea is that we have here a scintillator for energy spectrum, but we also have a fast silicon pin X-ray detector from Amtech, which we embedded in a LED box to get rid of the background noise and Bremsstrahlung noise coming from the beamline. And basically with the fast detector, we are able to detect basically every single event occurrence of X-ray production. And here you can see the basically schematics or what I explained so far. So the electron beam comes to the IP, it interacts with the laser, and actually you can see from the side view the laser has a very small angle, about 5 degrees. And obviously when you are able to synchronize perfectly at picosecond level the two beams, you get Compton X-rays, which then travel towards the detectors while the electron beam is dumped downward. So at the moment, we started very recently all this setup for the measuring of X-rays. We have not measured X-rays so far. We started a few months ago. We brought the electron beam to full operational values from low energy up to about 100 mega electron volt with full tunability of the spot size. And as of today, we are looking for X-rays. So we are basically setting up commissioning turns in order to start the measurements and with the detectors which we recently installed. Here you can see it was on the right bottom. Here was the first inauguration of the source of the facility, STAR in southern Italy. And I would so looking for X-rays. In the meanwhile, we have created a very great accelerator team, which for us, in my experience, was the most important key to actually get everything working and in a very short time. And so hopefully we can see X-rays soon. Thank you very much for your attention.
Thank you. Quick question. So how many copies of this, or my understanding that there are like 1.5 copies of this machine or something like that?
What do you mean copies? Like
or
in Italy?
Yes.
In Italy, it's the first one. It's the only ICS machine which started so far.
But Luca, I don't know if you're still here, Luca, but so Simon talks about the Star Africa.
Okay, Luca is actually
clarify
here.
for us, please.
Yeah, Lawrence, can I comment on that?
Yes.
Yeah. Well, there are ongoing pourparlers, ideas of developing and building a replica of STAR in Africa. The very recent news, besides the STAR Africa initiative that was pursued by Simon Connell in South Africa and the South African Center for Nuclear Energy, that was a good candidate to host a replica of STAR. There are very recent news that a memorandum of understanding was signed between ANFN and Université Mohamed Premier in Morocco, in Oujda, to develop a project based on a compact light sources in Morocco. This is very recent news. The present status of the Star Africa initiative is, I would say, not really known to me. I think it's something that was very promising, but waiting for some approval by some South African funding agencies. And if I can add a further comment, why nuclear energy center in South Africa? Because we know that these compact light sources can easily, quote unquote, easily be extended to nuclear photonic nuclear physics photon energy range. I mean, photons in excess of a few MeV of energy range. That opens the scientific case of nuclear physics. And if you allow me, I want to conclude my comment by complimenting Luigi for his very nice talk. I think one of the important points raised by Luigi's talk is the fact that these are compact light sources, where compact stays for compact in size, 10 meter, 10 million euros paradigm, but they are complex and challenging as bigger machines. I think you have seen by Luigi's presentation, you need a well-prepared and experienced acceleratorist team. You have to integrate many technologies, high power lasers, high brightness electron beams into a single machine, in an operating machine. And this is quite challenging. And I think that Luigi very well showed what you need in terms of human resources, expertise, and operational teams to develop and run these compact light sources.
Right. I was actually going to come back to that. I think one of the things our communities, both the African Light Source and the Greater Caribbean Light Source, is we have not engaged the engineering community as well as we could and should, because you're integrating a lot of different technologies to create an instrument or facility that is new in and of itself, right? My question is, how would you propose that we do that in a way that, or your center there, I guess there's these other compact light sources, sort of research areas at Arizona State. I know maybe a few others. Is the one in the Netherlands with what's the thing called? Smart Light?
Smart Light, yeah, in Eindhoven, the Netherlands.
Yeah. So how do we need to engage the engineering communities in let's call it the global South, specifically on these compact light source machines, because they present an opportunity to do sort of conventional synchrotron sciences, and then sort of, let's say for us, unconventional photon science with the nuclear structure and everything. Did that question make sense, Luca or Luigi?
Absolutely, Lawrence. And I don't want to step on Luigi's foot because he's the speaker, actually.
Right. I was just
but
saying.
Go ahead.
I can anticipate my quick answer. If you look at the experiences that you just mentioned, Smart Light in the Netherlands, Tomex in Paris, France, STAR in Italy, the Arizona CXFEL, and many others that we could, Tsinghua in China. You quickly see that you need to develop first the machine team, the acceleratorist team, clearly together with the scientific case and with the community representing the users of this user facility. Unless you have the machine team capable to design, deploy, commission, and then operate the machine, you are going to get into big troubles. Which is, by the way, I hope nobody's recording here, is one of the issues that we are addressing in South Italy, in Calabria, because the machine team is coming from an institution that is different from the host institution. ANFN in Italy in this moment is the only national research institution leading the expertise for building these compact light sources. but the host institution, that is University of Calabria, had to borrow or to buy this expertise from our national research institution. And you have a counterexample that is MuCLS, the Munich Compact Light Source, operated in Munich, in Germany, that was bought by a private company, Lyceum.
Yeah.
I think, Lawrence, you know
Oh,
and you
yeah.
remember.
Right.
Okay, if you look at their experience, they bought not just the machine from the company, from the private company, American, US private company, they bought also the team to run the machine. In the first 3-4 years of the machine operation, the machine was operated from California, Palo Alto, with the scientist in Munich, the Technical University of Munich, running the experiments with photon beam operated remotely by Lyceum. This is another counterexample that is relevant, I believe, because it tells you that if you don't have an accelerator team, you have to buy it. Either you develop by yourself in your host institution, campus, national research institution, whatever, or you have to buy. But if you have to buy, it costs a lot of money and time.
Yeah. Got it. So, what would a sort of compact light source workshop or school look like? And is there already one?
Well, we have a workshop actually in 2 weeks in Eindhoven. We are going to have, and actually Luigi, myself, and many other people will be attending. There is the symposium on what's the name, Luigi? Compact X-ray sources.
Yeah.
Okay.
ICXS.
Yes, the International Symposium on Compact X-ray Sources.
Compact X-ray sources, and this is a biennial symposium that gathers all laboratories and group worldwide working on compact light sources. And basically, we are going to have all the project that we have sort of listed, mentioned.
Okay.
European, Chinese. Yeah, also in Japan, there are a couple of them. And so this is a typical regular meeting. To the best of my knowledge, there never has been a school on compact light sources. And Lawrence, this may be a good idea or a good provocation, but so far, probably because it's a new generation, and so they are not yet that.
I see what you did there.
So okay, we get the idea. We'll try to to get along with it. Thank you.
Okay. Are there any other questions for the speaker? This is how you go about. Now, if I understood, you wrote a software package, or that you bought a software package that allows you to sort of simulate the control room.
We developed it in-house.
We developed it. Okay.
Yes.
So that must have some educational uses for sure, right?
Yes.
So we could probably use that somehow.
Definitely. There are many students, mostly PhD students, which are trained to improve it. So actually, so there was recently a PhD student who got her degree. So she's from the University of Calabria working on STAR. So most of her thesis was on Giotto, the implementation of the Giotto algorithm in STAR control system.
Okay.
But I just one quick comment. I agree with what Luca was saying. It's your choice. You either decide to save money at the beginning but do a lot of training. Students, so you make your own accelerator team group, which it's not very easy to find. I mean, from our experience, it's very difficult, especially to keep people, especially in southern Italy, which is more, let's say, it's not easy as the rest of Italy. So a lot of students which are trained, then they leave. So we are experiencing this issue. So we know how difficult it is to train and convince people, to be dedicated for an application. Or you can spend a lot of money and you can hire private companies who run the machine for you. I mean, this is done in many places in the world, like in Japan, the accelerator is run by Mitsubishi. So it's a choice. I mean, and we with Luca, what we are experiencing, we are pushing with the accelerator team building. So because we think that today's star needs a local team, accelerator team, which they started. So at the moment we are integrating. So we go there from Rome, from Milan, and but the goal is that in a few years, maybe in a couple of years, they have at least a minimum of between 6 and 10 people for all systems. As you said, from radio frequency, vacuum, electricity, water plants, which are not complicated things in a national lab. They become very complicated, especially in a university which is not used to this type of machines.
Yeah. All right. If there are no further questions for the speaker.
No, can I?
Yeah, okay.
Sorry, I just part of the team of Lucas.
Yes, Ilya.
And just add some, not comment, just add some information to what Luigi say. One of the important thing when you start build machine in your university, in your study, when you prepare student, such a machine like STAR, you educated student, and student not always finish with PhD, not going after postdoc, but they start be educated in accelerator physics. And one of the important thing that today we have a lot of medical accelerators, and we need medical technical people for manage this machine. And machine like a STAR is already start preparing people who can be technical. And if you start in university accelerator division, one of the important line is preparing technical people, technical personnel for medical accelerator for therapy, for hospital. And this is also a quite important thing, not just a source, not just accelerator, but also school, but not school for just for people to know knowledge, academical school for technical to manage medical accelerators. This I think quite important.
Okay.
Yeah.
So what is the objective of so that you did the MOU with the University of Morocco? So what is the objectives of that interaction?
That's for me, Lawrence, I guess.
Well, I guess you're all one team, right?
Yeah, we are one team, but I'm following the NFN president for the signing. Well, the focus is really developing a compact light source in the campus of University Mohammed Premier in Oujda. That is a city northern Morocco, in the Mediterranean section of Morocco. So the focus is collaborating, as any memorandum of understanding is a collaboration agreement, collaborating to prepare a project to develop a compact light source in the campus of UMP for medical application and also possibly for other, let's say, fundamental physics studies in QED or with high energy photons, and that can enrich the scientific case. The hope is that this project will benefit from co-funding from the Italian government Piano Mattei initiative that is an ongoing running initiative of our government to cooperate with the African countries in developing high technology project, developing of research infrastructure and many other things with the involvement, of course, Savasandir of the Italian industries and the Italian companies. So this is the way ahead. For the moment, we are just as the starting time. We have this memorandum of understanding that was signed between the president of the university, Moroccan University, and the president of NFN, and we are just starting today, I would say.
Gotcha. Okay. All right. Well, if there's no other questions for Luigi or any of the IFNM team, we'll conclude today's sessions for the conference, and we'll see you all tomorrow morning.
Thank you.
All right. Thank you, everyone. We'll see you tomorrow. All right. Bye-bye.
Thank you, Lawrence.
Bye. See you all.
Cheers.
Bye.